consider some of the most iconic biodiversity areas in Africa, so grazing lawns in the Serengeti. So there's a little bit of disjunct here where people take this idea of degradation and then apply it broadly without thinking too closely about the details. Now, Neumayer did think about the details. In his paper in 1975, he indicated that the vegetation attributes affect the shape of the consumption curve, and he did a lot of theoretical thinking around how the rate of consumption, the rate of growth under different consumption levels can affect the stability of these ecosystems. And at the same time, McNaughton, inspired by some work by Bell, kind of coined this term a grazing lawn. And in his paper, he said, a grazing lawn is a drastic reduction of canopy height and the activation of tillers that lead to a prostrate, dense canopy that then supports a lot of herbivores. So already, like many decades ago, there was this idea that heavy grazing doesn't always result in degradation. And many people picked up on this idea of a grazing lawn and did lots of exciting work showing how grazing lawns can increase nutrient cycling and can increase biodiversity, and they are unique and interesting habitats to consider. William Bond was particularly active here, and he also managed to broaden the ideas to thinking about two different ecosystem states that tropical grasslands can be in. A brown world, or a herbivore-dominated world, with short grass grazing lawns, and then a tall black world with tall tussock grasses. And in the 1990s, he and his students did a lot of wonderful research showing how these ecosystems differ in the trees and the birds and the grasshoppers and the spiders and the ants, and basically having both of these ecosystems in landscapes can increase biodiversity. So there was a general realization that these grazing lawns are perhaps things that we should be valuing instead of considering them degraded. But there was less understanding around exactly how we should be managing them and valuing them. Back in the early 2000s, there were lots of data coming out of Kruger showing that all the heavily grazed shortgrass ecosystems in Kruger were edaphically constrained. So in the Kruger National Park, there were shortgrass grazing lawns, but they were all constrained to nutrient-rich patches, like termite mounds or sodic sites. But at the very same time that that work was being done, in Vleughleum-Follosi Park, Joris Kromzicht and his supervisor, Jan Olf, did this like wonderful experiment where they just mowed a patch of tall grass, and the grazers came and they ate it, and 4 years later they had a grazing lawn. So I realized when I pulled this paper out, it's been cited 60 times, but I really think it should be more like 600 because it was really quite a novel and exciting piece of work. So there were these conflicting ideas around how these lawns are created and maintained. And that's, I suppose, where our project came in. There was recognition of the value of shortgrass ecosystems, and then there was this possibility that these systems could be created through positive feedbacks, that they aren't totally constrained by the landscape, that there's some kind of dynamic to them. And then there was also a recognition that Kruger actually has fewer grazing lawn areas than similar ecosystems. If you quantified the extent, it was about 15% in HIP at the time, down to about 2 to 4% in Kruger. And if you plotted across a mean annual rainfall gradient, the herbivore density, that's what you find in Africa. You find like a lot of herbivores at intermediate rainfalls, fewer in arid systems, fewer in mesic systems. And then the fire, the extent of fire, also follows this sort of unimodal distribution. But at higher rainfalls, you get more fire and less herbivory. And at low rainfalls, you get more herbivory and less fire. That's the pattern for Africa. But if you plot the numbers from Kruger National Park back in the late 2000s, Kruger National Park has this type of rainfall, but it had way more fire than you would expect, and way fewer herbivores. So the managers and the scientists in the Kruger National Park were bringing all this information and reading it and thinking, well, maybe there's something to be done to sort of increase the grazer habitat in the Kruger Park. And that was really the origin of the Satara Fire Grazer program. It was the brainchild of Kate Parr. She cooked up this amazing idea. And then it was supported by the wonderful people at Scientific Services, Dani Pinar, Navashni Govender, and Tertia Stradum. It was really a management experiment. We wanted to know, can we increase the area of grazing lawn in the Satara landscape of the Kruger Park? Those of you who know the Kruger Park know that it's this very open savanna with sort of very homogeneous grasslands, Bothria chloa, Thermida triandra, but there was an idea that perhaps this was a landscape that would once have had more herbivores. So that was the management side. But then we also wanted to know what are the consequences of adding shortgrass habitat to a tallgrass landscape. So what would happen to the herbaceous species composition, to tree recruitment, what would happen to soil processes? And then finally, what would happen to animal nutrition and demographics? Like, would we actually be able to increase the carrying capacity of the Satara landscape by adding shortgrass habitat? And that was one of the really key questions. And then the third thing that we had to address was, when are these shortgrass ecosystems desirable states that we want to manage for? So Kate's brilliant idea came from work that I had done with William Bond a while back for my master's, which had showed that fire, actually large landscape level fires, are active in taking these incipient lawn grass patches, which have high nutrients. And then when you have a large landscape level fire, all the animals that were grazing in these patches spread out into the lawn, into the fire, because the regrowing grass after the fire is really nutrient rich. So after a fire, herbivores can get forage of the same quality that they usually get in grazing lawns throughout the entire landscape. So they spread out and graze widely. And that basically results in tall grasses growing up without much intense grazing. And those tall grasses have really low nutrient quality. So that kind of entrenches this feedback loop where large fires prevent the initiation of grazing lawns. And that was really fun ideas, and we tested it, and we showed that when you have fire in the landscape, shortgrass patches don't persist for very long. Without fire in the landscape, you can maintain shortgrass patches for longer. But Kate's idea was like, well, if that's the case, if large fires spread herbivores out, why don't we burn really small fires and try to concentrate herbivores? So use this fire-grazer interaction to create grazing lawns. So that's what we did. We wanted to see whether you could use small fires to increase shortgrass habitat by creating concentrated grazing patches through the post-fire regrowth. And we had two wonderful master students, Drew and Zoe, and then we had an amazing field technician, Happy Mangena, who was there since the beginning of the experiment and basically managed and oversaw and created and added a huge amount of intellectual value to this experiment. It was massive. It was the first time I've ever done a landscape level experiment. So this is the Satara rest camp. And our experiment stretched across this entire burn block south of the Satara rest camp. It was a uniform grassland dominated by Thermida triandra and Bothria chloa. We tried out 3 different fire sizes because we didn't know how small the fires had to be. And we tried out 2 seasons of burn because we didn't know at what point the animals would be most attracted. So we ended up with about 27 plots, but some of the big plots we had to sample more intensively. So we had about 81 subplots to sample, and it was a massive amount of work. So that's basically, we started our experiment in 2013, and we applied an early season and a late season fire, and here's some pictures of the fires. You can see the type of landscape we were working in, not very attractive to shortgrass grazers. And we did that twice. We did it in 2013 and we did it in 2014. By 2015, we tried to apply the fires, and here's some pictures of us applying a fire in the early season burn treatment. But the grass had been kept so short that the fires actually didn't spread, so we had switched fire off in our experiment through this positive feedback with grazers. Now, you don't have to take my word for it, because Drew and Happy and Jason Donaldson did a lot of hard work demonstrating this. So basically, in our burn treatments, the mean proportion grass height, short grass height had increased about 9-fold to 90%, and all of the late season burns were failed ignitions. We couldn't burn the landscape. So we'd switched fire off in all of the late burn plots and many of the early season plots. We'd increased the short grass by nine times, and we had also tripled the bare ground cover. So remember that, because we took Winston Trollope to the site somewhere here, I think in April 2015, and he looked at me and shook his head and said, "Sally, what have you done?" How did we do this? Well, the fire grazer feedback that I indicated to you really did work. So over the time period of the experiment for the first 4 years, you can see in the control site, this is the accumulated wildebeest dung, but in the 2 burn sites, we had double the number of wildebeest, really. Here's some visuals. This was the landscape before we burned it. This is the landscape with the grazing feedback, and this is an exclosure showing how tall the grass would have been if the grazers hadn't been keeping it short. The wildebeests were sort of 10 times higher preference for the short grass, impala 5 times higher, zebra 2 times higher. So there was an increase in grazing. And that maintained this short grass ecosystem. And that's a picture of our late burn plots in 2015, an idyllic landscape packed with large herbivores and very easy to walk through and no ticks. But we were really interested in finding out more around the consequences of this treatment we'd imposed, and Jason Donaldson joined us for his PhD and did some amazing work on this site over several years. And one of the things he showed was that there was, as well as a structural switch, there was a compositional switch in the grass species that were on our shortgrass landscapes. This is an ordination, basically showing these plots over here were the plots that had been burned and grazed, and those were the control plots. And so with more grazing, we had a switch in species composition, and the traits of the grasses that increased when we added grazers to the system were moisture content and nitrogen content. So basically, when you add grazers to a system, you increase the amount of palatable grass and you decrease the amount of unpalatable grass, which is pretty exciting because that means we've now got an additional stabilizing feedback as well as just the structural differences. We've got a switch in composition to the types of grasses that grazers prefer. And in a way, it seems quite obvious, and this data has been shown many times. There are hundreds of papers all over the world showing when you add grazers, you switch the species composition. But the fact that you end up with grasses that are more palatable, so that grazing creates ecosystems that are more preferable for grazers, and likewise with fire, when you burn them, you end up creating ecosystems that are more flammable due to the species that are there, is quite astounding, actually. Cedric Solofonandrofata, a PhD student of Caroline Lehman, did this at a much bigger scale in Madagascar. She looked at switches from grazing to fire plots across Madagascar, and she came up with the same thing. You have these, when you add grazers to a system, you end up with these grazing adapted grasses with all these traits that make them more palatable. And she also showed, if you look across the phylogeny, that these grazing adapted grasses are very ancient, and the fire adapted grasses, there's no sort of clusters of grazing and fire adapted grasses. So there's some sense that there are these 2 grass communities that are both very ancient, and due to really interesting feedbacks, they create their own disturbance regimes. In the Kruger National Park, at our site in Satara, the insights that we got were that basically if you add grazers to an ecosystem, you can change the species composition in the same way that you would as if you went across many hundreds of kilometers across a rainfall gradient. Because across a rainfall gradient, as you go down the rainfall gradient, you increase the amount of grazers in the ecosystem. So if you increase the amount of grazers in a location in space, you switch the species composition. So we've got this idea that we're still hoping to test, that the turnover in life forms and in species that you observe across regional scale gradients can actually occur at a landscape scale in one spot, depending on what the dominant consumer is. So we're hoping to go and actually demonstrate some of these exciting ideas. That was where we'd got to in 2015, and then there was a drought. So this is a Google Earth image of our experimental sites during the drought. I think it was around 2016, early 2016. This was a really extreme drought in Satara. Satara was the sort of epicenter of the drought. I think basically from about April 2015 to the 16th of December 2016, we had practically no rainfall. And Jason and Happy went to the plots every month and basically measured bare ground, bare ground, bare ground. The tussock grasses cured totally. So there was no food around, and the animals all disappeared. It was a lot of hard work, but we did get some really interesting insights about what happened during the drought. The tussock grasses that had been grazed before the drought, we'd applied some fire experiments just before the drought hit, and the tussock grasses that had been grazed before the drought died. We had 90% mortality of those tussock grasses. On the control plots that hadn't been grazed before the drought, many of them survived. There was only 30% mortality. So this idea that it's the concurrence of grazing and followed by a drought that really waxed the tussock grasses was quite fascinating. And then the other thing that happened during the drought is that the herbivores moved away because there really was no food in that landscape. And they didn't come back for another year. So there was, once the rains came, there was a whole year of these ecosystems regrowing without any grazers at all, which was a wonderful test of whether we'd really created a shortgrass ecosystem because the grazers had gone. And so then we were like, well, have our grazed patches persisted despite the fact we have no grazers? This is a picture of a dead tussock grass surrounded by the amazing productivity of forbs that came up directly after the drought. So then after the drought, once the rains came back, as I mentioned, our experimental plots had high biomass. There was high biomass everywhere, but our experimental plots still had the different species composition. It was dominated by Urocloa mosambicensis, and you can actually see the line. This is the line where we had applied our fires in 2014 and 2013, and you can see the line just because of species compositional switches. We didn't apply any fires during the drought because it wasn't possible. And when the animals finally did come back, they did come back to our burn treatments again. Here's some pictures of animals having a field day on our Urocloa patches in the Satara landscape in 2018. But it was quite patchy. And we've been monitoring ever since. We had to agonize about whether to put in these 2 fires, but we decided to do it. So we reinforced the feedbacks that were already existing by putting in 2 more fires. And by the end of 2020, we still have this very strong difference between the control plots and the plots where we've been artificially increasing the number of grazers. We've been collaborating with Andrew Davies and Xenia and others to have a look at how this looks spatially. And so this is also December 2020, and you can see our burn plot has much shorter grass and much more uniformly short grass than the rest of the landscape. So, thank you. It's been 7 years that we've been applying this experiment. We've learned an awful lot, and we've taken our learning from Satara and expanded it to try to understand kind of grassy ecosystems and their biogeography across the whole of Africa. But did we actually create grazing lawns in the Satara landscape? Well, Gareth Henson, who is the grazing lawn guru, has this definition for grazing lawns, which I also embrace. He says, grazing lawns are short-statured, grazer-dependent plant communities that persist via positive feedbacks with grazers. So have we created grazing lawns? Here's some data on grass height from 2013 to 2019. In the burned plots, it took us a few years, but then we did create short-statured plant communities. They were definitely grazer-dependent, because without the grazers, they were this height. So yes, short grass, yes, grazer-dependent. Positive feedbacks, I think we have demonstrated some positive feedbacks. We've demonstrated that you can create these short grass systems just by changing the grass structure. You don't need any edaphic or sort of soil differences. And we've also created this positive, we've also identified the positive feedback through switches in grass species composition. So we do have the positive feedbacks. So we have got grazing lawns, in my opinion, and I hope in Gareth's opinion. But for how long do we have grazing lawns? In 2020, we still had grazing lawns, but we haven't collected data in 2021. So when we go collect data now, perhaps it will all become a tall grass, thermida-dominated landscape again. Which prompted us to start thinking around the scale at which grazing lawns happen, the spatial scale and the temporal scale. So what I've done here is I've plotted all the potential causes of a short grass grazing lawn ecosystem across a scale of size in hectares, and this is a logarithmic scale, and persistence, like how long we expect these sites to persist. So an animal carcass can create a nutrient-rich, heavily grazed patch at a very small scale for a very short amount of time. So can a fallen tree. Rhino middens are a bit more persistent. Termite mounds last a long time. At the large size, you've got hippo lawns, which can be really extensive. Communal rangelands can be very extensive. And this is where I've put our little small fires. Our small fires are not very big, and so far we've managed to make them persist for about 10 years. But they're definitely in the same realm as many of these grazing lawns that we see emerging naturally in these ecosystems. But we also have these really persistent lawn grass formations that are controlled by geological formations. So sodic sites are hundreds of millions of years of soil formation. And the gabbros around Orpingate have been around again for hundreds of millions of years. And as far as I can tell, those gabbros have been grazed heavily for hundreds of millions of years. There's no reason why they shouldn't have been. So some lawns are incredibly persistent. So were these lawns a good idea? We've shown that we can make them, and we've shown that they are lawns. But have we degraded the landscape around Satara, or have we rehabilitated it? I mean, Happy is clearly very pleased by the, and I'm very pleased. We go around congratulating ourselves, but we should actually be able to check what types of ecosystem functions have changed on the lawns that we've created. Especially in the context of the general way that people define degradation. You know, the assessment on land degradation says that the deviance of biomass from the local norm may be taken as a measure of land degradation. So we have definitely reduced the biomass at Satara. So according to the Global Assessment of Land Degradation, we have degraded the landscape. So let's see what we've actually done. As I've mentioned, we did kill all the tussock grasses. That was helped by the drought, but by 2017 on our burn plots, there were no more tussock grasses. We have found, when we looked at the species composition of the herbaceous forbs, we found a few lawn species that increased dramatically in our sites, and we found one bunch species that decreased. But we've only really applied these treatments for 6 years. So the sort of response of these long-lived forb communities might take many, many more decades to really come through. So the lawns increased the beta diversity of the landscape, but mostly due to having different species, not from being more diverse. They actually had fewer species. Our plots had fewer species than the tallgrass landscape, but they were different species. What about the trees? We didn't know what we were going to find there, but Mike Voisy did a wonderful study showing that these shortgrass ecosystems attract browsers as well as grazers. If you look at body mass of browsers, all the small-bodied browsers had high preference for our shortgrass ecosystems that we made. And then he also showed, by putting out some little seedlings and monitoring their survival, that our shortgrass patches had much lower survival. This is with an exclosure, that's without an exclosure in the shortgrass landscape. That's the tallgrass. So when these shortgrass ecosystems also reduce the establishment of woody species. And they also, he showed, reduce the growth rates of the woody species. So there's positive feedbacks which reduce tree cover on lawn and maintain the open ecosystems. During the drought, we monitored the juveniles and the species composition of the trees before and after the drought. And we found a massive shift in the juvenile species and a lot of new species emerging that hadn't been there before. So we got increases in 8 species and decreases in 4 species, and that was more extreme on our shortgrass grazed sites. And that work was done by Felix Trotter. I suppose the really interesting one in terms of our nature-based solutions discussion on Friday is what happened to the biomass and the soil carbon. So the above-ground biomass decreased dramatically. I've shown you that already. The below-ground biomass there was a decrease, it wasn't significant. So there was no significant difference in below-ground biomass in our heavily grazed versus our control plots. But there were more root gaps, so there were more patches of open soil where trees could establish. And then in terms of the sort of soil responses, I did this work with Pesheya Dlamini and his master student. Carbon decreased about 24%, nitrogen decreased about 30%, And then many of the other micronutrients increased on our lawns. And I think we should really be doing some more detailed studies of what sorts of carbon decreased, because there's been some wonderful papers published recently saying that even if heavily grazed landscapes decrease carbon, the right sort of carbon increases. But I'm perfectly happy with the idea that carbon decreased on our lawns, because all these other interesting things happened as well. So this is something we should discuss on Friday. So to conclude, what we've shown is that once a shortgrass landscape is initiated, there are positive feedbacks that come into play to maintain it. So the one positive feedback is with grass height. Short grass is more palatable than tall grass. But then there's also one with species composition, because grazer-adapted grasses are more palatable than fire-adapted grasses. And then at a landscape scale, once you have enough short grass in the system, then the fire gets switched off, and that prevents fire spread. We also showed higher browser densities reduces woody establishment. And then there's the other feedback, which I'm afraid we didn't test, but that's what McNaughton raised at the very beginning, which is that you get increased nutrient cycling rates and productivity. There are homogenizing processes that operate. Droughts decouple grazers and homogenize the landscape. High rainfall years overwhelm the grazer control. We showed that grass compositional response changes within 3 years. Soil carbon changes within 6 years. We had forb responses, but that probably takes much longer. We have data on termites and microbes and birds, but that is still to come. But we persuaded Kruger that it's maybe worth giving this a try at a management scale. So last year we initiated a management scale version of this experiment in Lower Sabi, where we applied these small fires in a landscape that management recognizes as having too much fire. And we're trying to create these ecosystems at a scale that's actually relevant to what the managers find useful. I just want to finish by saying that we found no signs of permanent degradation, despite the massive responses. Why not? Well, potentially because we were dealing with free-ranging herbivores, and you must see Mike Voisy's talk later to think about how this works when herbivores are constrained. But in that case, how do we distinguish degraded systems? I don't think deviation from local biomass norm is a sufficient definition when there are alternative stable states within the same, and each of those alternative stable states have different biomass levels. So I think I would argue that global assessments of degradation are biased against ecosystems with herbivores in them. And then that puts these ecosystems at risk of real and irreversible degradation and land cover change. I'm going to skip that because I would like to spend some time saying how wonderful it's been to work at the site with such an amazing range of colleagues. And thanks for giving me this opportunity.
Thanks, Sally. You've used your time perfectly. I had a few questions, but I'll catch you in tea time. So I think Sally did a very good job in introducing Michael already, and it's probably very well timed that you are now going to follow on from this talk. So thanks, Michael.
Morning, everyone. Yes, it's definitely a privilege to follow after Sally, who's inspired my project, along with a few others, including Michelle and Gareth and Matthew Harris. So I think we can all agree, and we're all aware of the diverse herbivore guilds that we find in wildlife systems. And these herbivores have a really important ecological and evolutionary role in these systems. In turn, they influence and impact a number of important ecosystem processes ranging from fire regimes, nutrient cycling, as well as influencing habitat for other organisms. And we can go through a few of their different traits, ranging in body size, water dependence, their gut type, their diet, and of course these all, in accordance with their abundance, influence how plants and vegetation communities respond to this herbivory. But a really important and widespread change in this this guild of herbivores has been the replacement of wildlife with livestock. And livestock are functionally depauperate, comprising mostly grazing cattle. So yeah, we were very interested in this topic of what are the consequences of the shift in herbivore regime. And we ought to really know what these consequences are, considering how widespread this change in herbivore regime is across the continent and obviously elsewhere in the world. And I think a lot of these comparisons that have been made between wildlife and livestock have taken place in very controlled experimental settings, a lot of them taking place in East Africa. And we noticed that there's a requirement and a need to compare very heavily grazed cattle systems, notably communal rangelands, with heavily grazed wildlife systems too. And this is where the grazing lawn idea comes in. So we designed a study in the Kruger National region of the Lowveld, so not too far from here. We had 3 sites in Kruger National, Kingfisher Spruit, Orpen, and a site in Manuleti, and then 3 sites in the communal rangelands near Slivokane, namely Gothenburg, Shorty, and Athol. We focused these sites all on the gabbro geology, which generate these fertile clay soils that attract herbivores and grazers. An important aspect of the study was to control for rainfall and try and keep the sites as close together as possible. So rainfall across the region is very similar. But of course, the management goals here are quite different, with Kruger managing for the full range of biodiversity, whereas communal rangelands really try to maximize the cattle in their individual cattle camps. So an important thing to be aware of is that in these rural and communal rangeland areas, a typical village will have access to one grazing camp, and that camp is then used all year round. And that obviously adds a dynamic to this whole assessment. So just to compare the different herbivore regimes and herbivore densities across the sites, which just sets the groundwork for the work. Across the 3 livestock sites, we had high cattle densities ranging from 0.6 to 1.6 animals per hectare. And then just to make sure we're comparing apples with apples, we conducted dung count surveys across the 6 sites. And just to really show the important bit here is just to really see that all of the sites were heavily grazed, even the wildlife ones, comparable to a communal rangeland, which is quite interesting. And again, what I mentioned about grazing seasonality, and this also feeds into Sally's idea of degradation, that the communal rangeland areas, cattle graze all year round. There's no rest from grazing, and so in turn leads to grazing throughout the year, whereas wildlife tend to move off these heavily grazed areas. And of course, communal rangelands have their own grazing history. We know that the sites that we chose didn't or weren't cultivated in the past, but they obviously have their unique grazing history going back in time. So our study aim was to assess how herbivore land use affects the herbaceous community, both directly and indirectly by the effect on tree cover. So the different herbivore regime types, wildlife as livestock, can directly influence the herbaceous structure and function by their direct grazing effects, or they can indirectly affect herbaceous structure and function by altering tree cover in different ways. So we know that livestock systems lack browsers, and in turn woody cover tends to increase in these systems. So our specific hypotheses were that livestock areas were higher tree densities than in wildlife areas, setting the foundation for the second and third hypotheses, that away from tree canopies, a canopy is basically just a zoomed-in version of an increased woody density area. So we're looking at the canopy scale, and we predicted that away from canopies, herbivore regime will have little impact on herbaceous structure and function. And then beneath tree canopies, we would start to see this shift in herbaceous structure and function. And that's largely because these cattle areas do graze uniformly across the landscape and aren't as impacted by things like fear of predation. And then lastly, we expected grass species composition to be fairly similar, and that four communities would differ due to this change in browsing and the lack of browsers in livestock areas. So the graphical representation of these hypotheses, low woody cover, but beneath trees, we expected taller grass and unique areas beneath canopies, whereas in livestock systems, we expected uniform grazing with elevated tree cover. And so we expected these areas to be similar across the two systems, whereas beneath canopy areas to be different. So our methodology was fairly simple. We had a transect sampling approach, and these transects radiated out from point sources and areas of high utilization to areas of low utilization. In total, we had 450 plots per treatment, 150 at each of the sites. And our field measurements were percent cover and species ID of grasses and forbs. We looked at species level traits for grasses and forbs, namely their life history, the annual, perennial, and then also their growth form, whether they grew upright or prostrate on the ground. And then we also took grass height, calculated tree density, and the presence absence of a canopy. Analytical approach was very simple. We just used mixed effect models to test the effect of land use or herbivore regime and canopy on various metrics of structure and function. And then we used CCA as a multivariate analytical approach for species composition. So our first result is looking at tree density across the two herbivore regime types. We found that in livestock areas, there was more than double the woody densities of livestock areas. And I think that's the real take-home message for this result. The other interesting thing to note is that with increasing cattle density, we didn't necessarily have an overall increase in woody cover as one might expect, but we did have an increase in the number of smaller woody plants at the areas of highest cattle density. And again, this is all well known and well documented in the literature that heavy grazing and absence of browsers leads to woody thickening. For our second and third hypotheses, I've shown here the significant effects above the graphs. And for grass height, this conformed to our prediction that away from canopies, we have very similarly heavily grazed shortgrass areas, and that beneath canopy areas and wildlife areas, we have much taller grass growing, whereas in livestock areas beneath canopies were still heavily grazed. And then for bare ground, it was less of a response to canopies, but more just a general treatment effect that under a livestock communal rangeland regime, which are grazed year-round, we have increased bare ground. And again, this is probably a result of the year-round grazing. I mentioned those direct and indirect hypotheses, so this I would categorize as an indirect effect of herbivore treatment, whereas bare ground direct effect, directly due to land use. And for our grass functional traits, very similar to our grass height result, conforming to our prediction that away from canopies we have similar stoloniferous grass cover, which is a trait that does typify these grazing lawn environments. And similarly, underneath trees, livestock areas have a similar proportion of grazing tolerant traits, whereas beneath canopy areas for wildlife, we have the lowest proportion of stoloniferous grasses. And then, similarly to our bare ground result, the cover of perennial grass cover was a direct result of herbivore treatment. And again, an indirect and direct response to these treatments. And then finally, for this part of the analysis, we looked at forb functional traits, which is something that gets not very much attention compared to grasses. And we found that annual forb cover and prostrate growing forbs were all greater in the wildlife treatment. And this is possibly due to elevated browsing pressure on forbs in these systems. But we also know that forbs are quite eruptive and sporadic in their life history. And again, these two both direct response to herbivore treatment. And then when looking at grass species composition, there are a lot of species here, which was a great learning experience for me in the field. But what we found was that these grass species composition across the two herbivore treatments pulled out quite clearly, which was not what we expected. One could go into the grazing history of these areas as a potential reason for this. But yeah, your typical Eurocloa grazing lawn species seem to occur in both systems, and some surprising species occurring in the two different treatments that didn't occur in the other. We found a lot more perennial grass species at the species level in wildlife areas and a lot more annuals in the livestock. For forbs, is another interesting one. This is just for annual forbs. There was very little teasing out across the treatments, and again, probably a consequence of the eruptive dynamic of forbs, and particularly for annual forbs. But in total, 44 annual species, 26 unique to wildlife, 17 unique to livestock, which was a very interesting result for us. The perennial forb species composition pulled out more clearly than annuals, which does make sense. These could indicate more of a browsing response, considering they're in the landscape for longer, persisting for longer. And again, this could be a response to the browser guild occurring in wildlife areas, but not in livestock. The livestock areas that we worked in had very low goat densities, so I think they had very little influence on the composition in this landscape. So to conclude, through all of these results that I've showed you, we found high woody cover due to heavy grazing and browser absence in livestock areas. And I think this elevated woody cover has an important influence on the herbaceous community, and through some of the results, such as the grass height result, stoloniferous grass cover, those are all impacted indirectly through the herbivore treatment. So wildlife and livestock have direct and indirect effects on the herbaceous community. I think these then translate into species compositional differences, which we've seen in the last two results. And then I think an interesting thing to note is that these two systems do appear to be actually quite unique. The livestock areas, which are on very similar soil, similar rainfall, are actually quite unique forb species. They appear to have unique grass species. And so I think it raises the question, that these aren't just subsets of wildlife systems, and are our cattle systems creating unique and novel environments? And should we be protecting those or not? I think it's something worth considering. And then on a final note, I've discussed aspects of cattle management, like seasonality of grazing, other things like herding style and kraal size in these environments has a profound influence on what cattle do to the landscape. And I think when considering the impacts of cattle and livestock on savanna functioning, these aspects really need to be taken into account, because they do represent the realities that we find in these cattle systems. Thank you very much.
Thank you so much, Michael. You have used your time, but our next speaker is not here. So if there's a question for Michael, we can take it. Okay, I don't see any hands. Then we're going to, sorry, Susie?
Thank you. That's a really fascinating result about, the difference in grazing, in grass composition between the two treatments. And remember Daisy, she looked at something related, and we did it on the granites. And the grass composition in Magnoleti and Welverdiend, it was like chalk and cheese. I mean, it was hard to believe it was the same vegetation type. But I wonder, you sort of attribute this to the seasonality and so on, but could you maybe say something about the possible mechanism, like why the grasses you find in the wildlife area are different, and what especially makes the particular grasses that you find in the heavily continuously grazed system what they are? What attributes do they have that allows them to cope with that?
No, it's a very good question. And I think it's something I've struggled to try and understand and interpret through these results because there are so many factors. Grazing history, for example, and going back an extensive period of time back, I wonder if these areas have been supplemented potentially, and so species have come in from elsewhere. We found a few non-indigenous grasses in those areas. So I'm wondering if those have come in through cattle feed potentially. I guess dispersal of grasses between areas. We've got, in wildlife areas, we've got animals moving greater distances. And I'm wondering if in cattle systems, which are just so focused, and animals not moving out of there, whether that doesn't allow for dispersal of grasses. I think another aspect which could influence is fire, the presence of fire. So you might have more fire grasses in your fire-adapted grasses in your wildlife areas, because obviously livestock, because of the seasonality of grazing and the fact that these guys don't have more ground to graze the cattle, they don't burn because they're afraid to burn. If they do burn, then they potentially burn the fodder for their livestock. But yeah, maybe we can share a beer and discuss it.
Thank you, Michael. So we're going to move on to Jan Kruger, who is going to stay with the same theme and tell us about grass species composition and large herbivores. Thank you.
Okay, hello everybody. Thank you for coming. It's a big pleasure and honor to be here on such a nice conference. And today I would like to talk about the relationships between large herbivores, grass community composition, grass richness, and grass abundance in Kruger National Park. I would like to thank all colleagues from our team that were included in this research, and also my home institution, Institute of Botany of the Czech Academy of Sciences. First, few brief notes about grass-herbivore interactions. Herbivores are crucial for maintaining grasslands in savannas because they change competition relationships between woody species and grasses. And grass is a fundamental food source for grazers and mixed feeders because it's their main food. And what's important, that herbivores in savannas affect grass abundance and richness, and vice versa. So that also grass abundance, richness, and patterns can affect herbivores on the other way around. Here in Kruger National Park, grass abundance and diversity is driven mostly by precipitation, soil characteristics like soil depth and nutrients, and also by grazing. In this talk, I would like to focus on the interactions between herbivores and environmental settings and these effects on grasses. In particular, I would like to speak about relationships between grazer abundance and richness and grass species abundance and richness under differing water water and nutrient availability. So, to the methods. This research was a part of the bigger project, which is called Mosaic, monitoring savanna biodiversity in Kruger National Park. And basically, we monitor different groups of organisms and their interactions. Experimental design is as follows. We established 60 permanent plots, 50 times 50 meters in size, in 3 habitats that should reflect water availability. So the plots were located at crest by seasonal and perennial rivers, and also on 2 bedrocks, granite and basalts, that also should somehow reflect nutrients at site, because granites are, or soils that are derived from granites are poor, and basalt-derived soils are rich. Basically, we estimated cover of all plant species in plots. Here I will talk only about grasses, because they are of interest. And we also monitored occurrence of grazers that were recorded by camera traps. In this talk, I will focus only on ungulate grazers, so these 13 species, and they are grazers and mixed feeders, and I will call them just grazers because these both fielding goats feed on grass, and they affect grass. Data were analyzed by linear and generalized mixed effect models, where grass covers and grass species richness were the response variables, and also by multivariate methods for grass community composition. So for for covers of particular grass species. So what we get for grazer abundance, just briefly, I would mention that the most common herbivore was impala, and this picture also shows the differences in feeding goats. Species in bold are grazers, those in plain form are mixed feeders. And also I wanted to say that this picture shows the proportion of habitats, and it's clearly visible that most herbivores or grazers were recorded at perennial rivers, about 2/3, and the least at crests, about 7% or so. For analysis, we excluded 3 rarest species. And now to the results of generalized mixed effect models for grass cover, which was response variable in here. So we had 2 models. First for grass abundance and other variables, and second one for grazer abundance, and the second one for grazer species richness and other variables. So basically, we got significant results for grazer abundances and habitats, and I will talk more about these results further in detail. There was no significant relationship for grazer richness, and no significant interactions, as you can see. So in detail, if you look at the relationship between grass cover and grass abundance, grazer abundance, so grass cover is decreasing with grazer abundance. For habitats, the least grass was at perennial rivers, and the other two habitats didn't differ in grass cover. And for bedrock, there was more grass at basalt bedrock, but these differences were not significant. Now to the grass species richness, again two models, one for grazer abundance, the second one for grazer species richness. And you can see that it's different. These are results of linear mix effect model. And there were no significant effect of grazer abundance or species richness alone, but marginally effect of habitat and bedrock. And what's the most interesting here is that there were significant interactions of grazer abundance and bedrock. The same for richness, and also the interaction with habitat was significant. So again, in detail, for grazer species richness, grass species richness, sorry, the most species we found on crests, and this relationship between richness and water availability was sort of decreasing towards bigger water availability. Then there were more grass species on granites than on basalts. This difference was significant. And this next slide is about interactions, which I personally find most interesting. And you can see relationship between grazer abundance on different habitats and bedrocks, and its effect on grass species richness on the y-axis. So you can see that this relationship is positive on crests. So with increasing abundance of grazers, the grass species richness also increases. And the relationship between these two is sort of flattening with water availability. For bedrock, there was a positive relationship at basalts and the opposite one on granites. There is similar picture for grazer species richness on the x-axis. The other variables or the other things are exactly the same, and the results are also very similar, just with one difference, that for seasonal rivers, the relationship between grass species richness and grazer species richness is decreasing. So now to the results of grass community composition. We tested similar factors as in the univariate models, and so it was grazer abundance, grazer species richness, and also abundance of individual grazer species, and none of these predictors was significant. So there was no relationship between grass, in the covers of individual grass species and these variables. To sum it up, grazing pressure leads to a lower grass cover. In this system, disturbances by animals reduce grass cover and can also exclude some species that are intolerant to disturbances, like water disturbances by trampling or grazing. High nutrients can cause dominance of a few grass species, and grass species richness is supported by grazer abundance, under high nutrients. So this is probably dominance control of some grass species that dominate in the ecosystem. And we got similar relationships for grass species richness as for grazer species richness as for grazer abundance. So finally, some take-home messages. Relationships between abundance and richness of it's important to highlight that some of these relationships can have bi-directional explanation. For example, for this pair of variables, herbivore species richness and grass species richness, it can be affected both sides, that grasses can affect animal presence by attracting them by bigger or more various food supply, and also grazes, of course, affect grasses, as I said. I would like to point out to some related talks from our group, botanical talks, and it will be talk of Klára Pišková, which will talk about browsers and shrubs. Petr Pišek will talk about invasive plants in Kruger, and Martin Hejda about overall patterns in plants. So I would like to thank you for attention, and also thank our guards for being us in the field.
Thanks so much, Jan. We can take one question, if anyone has. Over there, Sally. Can we get? Don't worry, Sally, I'll bring you.
Thanks very much for the very interesting data. It's really great to see across the whole park the patterns. Is it possible to click back to your graph of the herbivore biomass, grass height, or grass biomass?
Already another presentation, sorry.
So you basically had herbivore biomass on the x-axis and grass height on the y-axis.
Yeah.
And you were like, increasing herbivore densities decreases the grass height. But what I took away from that graph was that you only get herbivores in systems where the grass is less than 10 centimeters tall. So I was wondering, can we assign causality? Like if you find herbivores in shortgrass areas, it could be the grass is attracting the herbivores as well as the other way around.
Yes, exactly. As I said, these relationship, I think that they can be both ways. So it's sort of correlations, not causal relationships. You are perfectly right. Yeah.
Okay, thanks so much, Jan. And then we're going to move on to our last speaker in this theme, Peter. Thank you. Peter will tell us, how do you estimate grass biomass using LiDAR?
Morning, everybody, and thank you so much for having me here. I'm Peter Boucher. I'm a postdoc in the Davies Lab at Harvard University, and I'm going to be showing you some initial results this morning from a study we're doing on mapping grass biomass with high-resolution UAV, or unmanned aerial vehicle, LiDAR data. And right here, you can see some of the data that we're working with. This is a side view of a point cloud, so it's millions of points, each with an X, Y, and Z coordinate that really define the vertical structure of vegetation in an ecosystem. And this is actually from the Buffalo Camp Exclosure right here. You can see the fence right there, and it's colored by height right here. So you can see that blue is lower, red is higher, and you'll see a few more of those. So when we have this amazing data, there's always this idea to sort of make up new metrics and see how far we can sort of push it to measure and map different parts of the ecosystem. And so one of the things that I really wanted to do with it is to try to map grass biomass at large extents and at fine scales, and I'm talking about a meter squared across a large area, so about 1,000 hectares. And this is traditionally done in the field with a disk pasture meter. which has a great relationship with grass biomass, but it's really just a point measurement. And as a result, you miss a lot of heterogeneity, and you can't cover as large an area unless you have a huge field group. And would be so many great applications for this kind of work, for a wall-to-wall map, essentially, of grass biomass, if we can get this to work. We have fire modeling, fuel loads, grazing, and tree-grass interactions, just a couple of them. There's many more. I'm sure you guys can tell me more than I can tell you. And so our real aim of this study is to take this new technology, LiDAR from an unmanned aerial vehicle. And what LiDAR does is what LiDAR does is it's like sonar, but with light. So it essentially sends out pulses of light with a laser that bounces off objects in the environment and comes back to the instrument, so it records it. And it does that millions of times as you fly over an area, and you get a point cloud that looks like this, colored by height. And these LiDAR-based models have been used for woody biomass for a long time, shown great relationships. So why not also above-ground grass biomass? So the instrument we're working with and the sensor package we're working with is called HALO, the Harvard Animal Landscape Observatory. It's integrated with a thermal camera, a regular RGB camera, and also a fantastic LiDAR instrument. And what this instrument allows us to really do is to get through the grass layer, get through very small gaps in the grass layer with very high pulse density. And that's why this is not something you would really do before with LiDAR. But now you can fly so close to the ground, your laser beams as they go down are much smaller, about 3 centimeters when you get to the ground. So you're actually passing through the layers of grass. You're recording the ground, and you're also recording the various blades of grass as you come through. So you're getting a profile of the vertical structure. And you can do this across a large area. It's about 1,000 hectares per day, give or take a few hundred. And so that data is wonderful, but what you really need is field data to do this, lots and lots and lots of grass plots and clippings. And so we tagged along on another project we were doing and Sally talked about this morning, this site at Lower Sabi. And we managed to get some grass clippings, about 96 within each of the plots before they burned. And you can see here's just a histogram of grass biomass. You can see it's pretty uniform here. Everything's kind of around the 100 to 200 gram range right here in the middle. And so we did 96 plots here. These are in the southern basalts, dry season. These are the grass species right here in order of frequency. The other site that we have that we just recently collected in January is at Impala Research Center up in Laikipia, Kenya. So right over here, north of Nairobi, northwest of Mount Kenya right here. We only have 30 clippings from this plot, but we have clippings from all over Impala that I still need to process, but this is just an initial set. And what we did here was we grabbed sort of a range of biomass here, so going from 0 all the way up to sort of 400 or even 500 dry weight. And a big difference between this site and the Lower Sabi site is that we actually marked those locations with flags so we could see exactly where they were in the data, whereas at the Lower Sabi, we just used GPS, differential GPS, which was high accuracy, but that'll come in later. And these are the grass species too. So when we're actually trying to pull out some of our LiDAR metrics from this data, right? How do you turn a cloud of points into something that's usable? Well, one of the ways I was thinking about modeling grass biomass is it's a product of geometry and density. Pretty simple. It's basically what's the volume, or what's something that describes the volume, like height of this cloud, and also what's the density of it. And so to describe sort of the geometry of it, we grabbed a bunch of metrics that had to do with heights, mean heights, percentile heights, going all the way from the bottom to the top, max height. We also did variation, and then also grabbed a volume metric by wrapping a convex hull around this. We used this cover metric And this might not be a typical metric that you're used to when you hear about cover. It's a vertical profile of cover derived from LiDAR data. So when you're flying over something like a tree right here, you have some pulses that get hit on top and produce a point, and some that go all the way through the canopy to reach the ground, right? And if you use the ratio of those pulses as you go through the canopy, this right here is the number of hits by height. So you're going down. You have about 800 pulses total within this tree. And say at 10 meters, you have hit an object 400 times with those pulses, or 400 of those pulses have hit something at that point. That would mean that at 10 meters, you have 50% cover. And so you can do that in whatever height interval increments you want. It just depends on the density of your data. And so we did it at 10 centimeters. And what you get is sort of a cover profile right here going all the way to ground. And so this tree is just for an example. What we're really interested in is this right here, right? So when I'm showing you cover profiles, I'm talking about the cover of the grass layer right at the bottom right there. But what's really cool too is that you can do this underneath trees. Since it's a continuous metric, you just need to know where the grass layer starts and ends. This is a pretty typical thing that you do to get plant area index, just FYI. So we ran some feature selection because we have all these variables, about 36 of them, and used the lasso regression for that. And so this is just an initial crack at a linear model. And we came out with a few important variables. First one being percent cover seems to be the most important. That's why it pops up here with a really high beta coefficient. And then we have these mean and max heights seem to be pretty important, but kind of equally important between mean height, max height. And then also variation in height seems to also be important too, which all makes sense. So when you just create a simple model just right out the bag, using cover at the very base of the ground, so at 0 meters, and then also mean height right here, and these two x variables are plotted right here with grass biomass on the side right here. You see a pretty straight line with our cover metric, which is great. That's what we want to see. Mean height does pretty well too, but there's definitely something nonlinear happening here that we need to account for. And when you look at our actual residuals, you can kind of see that we're doing an okay job, but there's something funky going on here, right? What we're doing is we tend to be over-predicting the lower biomass and under-predicting the higher biomass sites. And we get an R squared right here for explaining about 66% of the variation, about 60 grams right here, root mean squared error, which gets to about 22.5% error. And I should say too, we did this for half a meter by half a meter plots right here. So pretty good for a high spatial resolution, I'd say. One thing that's really interesting and has kind of stumped me, I'll be honest, is that if you just include the Impala data, so the data from Kenya, the model performs much better, at least it has a much better relationship. And your error, goes down a bit. It's not a huge reduction. But it does do much better, and actually your parameters are a bit better too, or your parameters are pretty similar between both models, but it just seems to fit the Impala data a lot better. Thank you. And that's the predicted and the field right here, a much more even spread than this combined model. And so that's a really interesting thing to me, is that why are we explaining grass so well at one place, whereas something like this cover metric is not really doing a very good job at Kruger right now. And so there's a few options. There's many options for what could explain the difference between these sites, right? First of all, we do have geolocation error introduced. At Impala, we really had flags that could chop out the plots exactly where they were in the data. Whereas we just use a GPS, and even though that was quite high accuracy, things happen. Also, it's possible that there's this actual difference in plant functional traits at these places. There's certainly differences in species compositions that we've seen. Even though they shared a dominant species, they had, other than that, they had completely different grass species. So this is a great figure from Wigley et al., showing you sort of the difference between these tufted grasses, these mat-forming grasses. And so we may need to incorporate that into the model somehow, maybe even do an object-based detection like they're doing right here. I think that would be fun. And then also there's this other sort of material composition, is, you know, how dry is the grass, how much moisture is there, also what's mixed in with it. You know, also when I'm talking about grass here, I'm actually talking about every plant that was in the plot. So it could be a forb. There could be some woody material too that's growing in there, even though it'd be quite small. And so essentially we just need to get some more data to start to parse some of these things out. And something that I am really excited about too is that I think in terms of this last question of trying to understand sort of composition and the content within each of these grass layers, we may actually be able to grab that directly from the LiDAR data. So right here, what you see are these waveform metrics, which are essentially, they're kind of like reflectance properties that are measured by LiDAR. So we have two right here, deviation and amplitude. And I've just clicked out grass, a shrub, and a tree. And the color scales are all the same. So what you're seeing here is how different these variables are for each of these classifications. A tree looks really different from a shrub, which looks different from grass in all these cases. And we can definitely, I think, use this in order to make our model better and to also maybe understand what the composition is in here. And so that's pretty much it. That's a quick study. I'd say that our sort of first model worked pretty well and had a pretty decent amount of error for a small spatial resolution and a pretty good relationship. There's still a fair amount of work to do. We have to explain this variation between sites, try to understand that, improve our model, and try to fix some of these nonlinear things happening, and add some more field data now that we know a little bit about what works for this site. And I just want to say thank you to everybody who helped with this. There's many more names on here than I could include. This is a group of us last January who were out there clipping grass and flying drones in Impala. So thank you very much. I'll take any questions you have.
Thanks, Peter. So we do have time for questions, and I see one there at the back. Kobus.
Yeah, Peter, thanks for your talk. I think it's interesting, and there's interesting applications that can come from that. In my experience, that Pennisetum straminium dominated grasslands, especially on igneous soils, tend to be very homogeneous, both in terms of structure and cover. And that probably explains your better linear fit to those two models.
Great.
Thank you.
Great. So basically how homogeneous it is.
Is there any other questions? So Susie's got one, but while the mic goes to Susie, I was also wondering just whether you did it in the same season, because maybe when grasses are standing upright as opposed to kind of haven't fallen over, that might affect then the actual cover that's open. And when you did your clipping, did you take the grasses out, or was it grass and forb?
Grass and forbs. Yeah.
Okay.
So I was trying to clarify.
Okay.
Yeah. So they were both in the dry season, and I should say too that Impala was having a very severe drought at the time. They basically skipped a whole rainy season, so that could also account for it that they were pre-dried for us when we measured them.
Okay, great. Susie?
Just a very quick technical one. You said that cover at ground level was the best predictor of biomass. Is that the same as basal cover? What we would call basal cover, you know, the amount of cover.
I don't think so. I think when you're talking about basal cover, you're thinking more about a horizontal metric. Is that right?
No, it's literally where the plant touches and goes into the ground. So it's really at ground level. Where it's bare, it's, you know, I mean, the grass might dangle onto the ground, but it doesn't actually. We can talk at tea maybe.
Yeah, I think it might take some
specific
looking at
reason for
diagrams. Thank you, though.
Okay, then our next speaker, and sorry, Peter, thanks so much. Yeah, that was great, and that's going to revolutionize any field technician's job going forward. Kate, we've kind of moved you to the end of the session. Sorry, we're skipping over you. Kay doesn't want her talk streamed. So, so if you don't want your talk to be streamed, then please just tell us before the session, and we are just going to move you to the end of the session. So we will move on to Tony. Tony's going to tell us about herbivore exclusion and bush encouragement. Thanks, Tony.
Morning, everyone. Thanks. I had to moderate my title a bit to fit it in. But also, you know, when you think you got some results and then you write a title for your abstract for a conference, you give this really cool long title with, it suggests you got amazing results. And then when you put in the talk together, a few days before, you realize you got nothing to justify the title. So I had to kind of change mine to a question rather than a definitive statement. Anyway, before I start, let me just acknowledge many people who have been involved with this study. It's a long-term study from the Letaba exclosure in the north of the park. And it was an experiment actually started long ago in 2002, but Francie Sibbett from Northwest has been involved for a long time, herself and many of her students, in collecting much of the data I'm going to talk about. And then myself and others stay on staff, also involved there over the latter half of the experiment. And then, of course, many from Sandpark Scientific Services who have been involved in keeping this thing going, and particularly Corley and Judith in getting it going and getting some results out. Okay, so we know that elephants push over tall trees. There's tons of work done on that, lots in Kruger. The Letaba exclosure as well, it was included, for example, in that study. It was an airborne LiDAR study that really showed how tall trees are declining in the park, including at the Letaba exclosure. In fact, more rapidly at the Letaba exclosure than most of the other sites studied. But what I was more interested in with this experimental site is what's happening with the smaller trees, the shrubs and the short trees. We know tall trees are declining, but are elephants also playing a role in regulating the density of the smaller trees? Because as we know, bush encroachment is an issue over much of the savannas of the world. So the Letaba exclosure provided us a potential site to look into that. For those who don't know, it's located up along the banks of the Letaba River, drier part of the park, Mapani Veld. Not particularly exciting and hasn't attracted as much research as areas in the south of the park. It's the same design as the Nkutlu exclosure here closer to Skukuza. It's got a full exclosure excluding all herbivores, and then a partial which excludes just elephant and giraffe. There's also fire treatments worked in. And it was originally, the original question of interest was what are elephants doing in the riparian zone of the rivers. So it has a riparian section, but also an upland section away from the riparian. And actually, I'm only going to be speaking on the results from the upland because the regular flooding just made it, it didn't make it possible to look at long-term vegetation change in the riparian zone because there was repeated fence damages and plot markers getting washed away or covered by sediment. And the elephant density in that area is as high as it is anywhere in the park. Between 1/2 to 2 elephant per square kilometer based on the long-term census data. Right, so if you go there at certain times of the year, it's a pretty desolate-looking area. You might think you're in a war zone at certain times. This was at the end of a year of drought, the end of the dry season following that really dry year of 2015-16, and it just looks terrible. And yet these sad-looking plant communities do sustain a pretty large herbivore density. Not only the elephants, but hippo and a whole range of other herbivores that, as you know, are really common in these riparian river valleys. You can actually get some decent grass growth there. So this is a picture taken inside the exclosure. So with little or no grazing and with decent rainfall, above average rainfall, you actually do get some grass, and it looks a bit greener and more productive. That's just the design of the experimental site. So here we've got a full exclosure, and then the partial over there, 40 to 50 hectares large each. the riparian zone, alluvial-based soils, but most of the area on granitic-based upland soils, quite shallow, quite rocky, not particularly productive, and a mean annual rainfall of only just over 400 mils a year. So that's kind of what it looks like, or what it looked like, I should say, with the full exclosure. So inside, a lot more grass, taller trees. In summer months, actually, that grass biomass really quite impressive, the difference, at least on the alluvial soils in the riparian zone. On the outside there, we'd have to check with Sally and Gareth, but that might be considered a grazing lawn, which would be maintained by Hippo. On the upland site, not as exciting. It's a very sort of slow system, slow to change. But within the area protected from elephants, the mopane trees do get a bit taller. They're sort of hitting 4 or 5 meters. Whereas on the outside, this side is the outside of the full exclosure, they really are all maintained in a kind of hedge by the elephants. More than 90% of the trees have primary stems broken by elephant. They're really pretty thorough in just browsing the top of every single mopane and keeping them short. For the partial exclosure, that's just to give you an idea what it looks like. So there's the bottom half of the fence is not fence, so that herbivores can move through. Occasionally, young elephant would move through, and sometimes bigger elephant would follow them. But in general, elephant not going inside there. And then that obviously keeps out giraffe as well, the high electric strands. But giraffe are really not very common in this area. So we didn't think the exclusion of them had much effect on anything. Okay, so when the site was set up and built in 2002, a long-term vegetation monitoring design was established, sampled for the first time in the summer of 2002, 2003, and then about 10 years later, and then about 6 years after that again. So we got 3 points in time, pretty long time period, encompassing some really wet years, particularly in the first half, and then that really severe drought from 2016 to 2019 in the second half. Quite a thorough design. So for the plots that we could use for this analysis, we ended up with at least 30 plots of 20 by 10 meters in which all woody vegetation was recorded. And that's pretty thorough, particularly considering that the tree and grass shrub layer is pretty homogeneous in this environment. Also some dispasture meter readings were taken each time to estimate the grass biomass. And as I mentioned, data for riparian plots not included here. We also at a later stage, towards the second half of the experiment, we set up some camera traps just to kind of verify if the partial exclosure was doing what it was meant to do. And then we also wanted to get more into demography, so we tagged some trees in 2011 and we did annual resampling of those. The camera trap data, just briefly, it did exclude elephants. The partial was excluding elephants. That's what I'm trying to show over there. So this is just the frequency that an elephant was recorded on the traps, animals per month. And yeah, occasionally some in the partial, but in general a big difference. And perhaps more importantly is all the other species. So these are the most common herbivore species recorded by the cameras, and very little difference between them. So the partial wasn't deterring the other species, which is good. And then related to that, the grass biomass, massive difference obviously when you exclude all the herbivores, about 3 times more grass biomass within that full exclosure. Slightly more in the partial, although that wasn't statistically significant. These values, for those who are unfamiliar with grass biomass measurements, these are pretty low. I mean, this is an average across all 3 sampling times, but 250 grams per meter squared or kilograms per hectare is pretty low. All right, so that's the main result coming out. So this is taking all the woody plants recorded in these plots over time, and a bit of an increase, but not much in the control. In the full, surprisingly, not much going on. The density of trees and shrubs about the same, but then in the partial, wow, this massive increase from over the last 16 years. So when I got this graph, I realized I should have really gone for a 5-minute talk and ended here, because that's pretty cool. I mean, I think that justifies the original title. If you have no elephants, but all the other herbivores, you get bush encroachment. Wow, cool. So that explains bush encroachment over the semi-arid areas of Africa, perhaps even other savannas as well. Write it up for nature, done, awesome. But unfortunately, when you start to look at this a bit more closely, it's not that clear. And what's particularly worrying about this graph is this. So not a big, well, it's a fairly big increase from 2002 to 2018. Why in our control plot where we have all the elephants are the woody plants still increasing? What's going on there? And why in the absence of all herbivory and disturbance are trees not doing so well in the full? So if we look at that a bit more closely, okay, so this is a bit complicated. I'm going to go through just different parts. But starting off in the control, what was driving that increase over the last 16 years was really smaller shrubs in the control areas. So under 1 meter tall, a big increase in our control areas, matched by decreases of taller trees. So this is kind of what you expect with elephants. There's taller trees before the elephants sort of return to the area. And by their continuous browsing, they're pushing taller trees into smaller size classes. Perhaps that's what's going on. Although maybe this is also seedlings' genuine increase in density. We don't really know. But certainly some of this decline there could explain some of this increase. It's just pushing taller trees into shorter size classes. And when we look in the full exclosure, we also see some sort of things that we might expect, like there's the emergence of taller trees here, right? So there's hardly anything above 5 meters in the control plot. When you've excluded elephants, some of these shorter trees have become taller trees. We've seen this progression. But we are also seeing some increases in the smaller size classes. You would expect kind of decreases because the smaller trees would move up into the higher size classes, and if there's no recruitment, then there should be less. So here probably there is some recruitment seedlings coming through, but there's also some movement of medium size into taller height classes. And again, in the partial, we see increases almost throughout, and more so than in the control. So something's happening in the full exclosure that's not letting trees grow quite as much as in the partial, and that's probably competition from grass. That's my theory anyway. A lot more grass biomass in there, and maybe that's just preventing the trees from growing as fast. So when we look at the data from the tag trees, from individual trees that we set up in 2011, this is how they changed in height over time. So each line here is a different individual tree. We had between 12 and 25 in each of the treatments, and really flat. And actually, when you worked in this place, like going back year after year to remeasure these trees, it was kind of like working in a museum. Everything's just in the same place, the same little trees with their tags, and they've hardly changed, they've hardly grown bigger. It's just nothing going on, very slow system. Actually, in the partial, there was some sort of height increases going on as expected, and in the full exclosure, a bit of a mix. When we average those out, you do see actually, even though the numbers are small, that trees were growing taller, were growing faster in height in the partial, and slightly more in the full, and shrinking in the control. But these are really small numbers. We're talking about an average of 4 centimeters per year of height growth in the partial. Just for interest, okay, that was the dominant species, Mapani. The third most common species was the red bush willow, Combrinum apiculatum. And there, yeah, foster height growth as well, but also in the full, so maybe not affected as much by grass competition, and again, shrinking in the presence of elephants and all other herbivores. All right, then just to end off, there's always, there's like when you finished your talk and everything, and then you think, well, I still had that other data, maybe I'll just quickly analyze that and throw that in. So that's what I did late last night, and I really just messed up the whole title and everything, because this is, in addition to tag trees, every time we went to measure the trees, we would also record the presence of any seedlings, and these are proper seedlings, like actually germinated that summer with cotyledons present and so on. And what we found, there were just a lot more seedlings coming up in the presence of all herbivores. Most of these were Mapanis. Now and again, you'd get more interesting things like Acacia nigrescens. But the vast majority of seedlings were from the dominant species, Mapani, and just so many more outside the fenced area. And why was that happening? And so that can explain why we've seen this increase in the density of trees and shrubs in the presence of all the herbivores. They're still somehow, despite all the damage to the adult mapanis, still managing to produce a lot of seeds and recruit. How many of these go on to survive and become actual shrubs, we don't really know yet. All right, so just to end off quickly, so yes, it seems that elephants do regulate the density of the tree layer, but wasn't quite sure about that. I went with do for now, but you might want to change that option depending on your interpretation. And also with over time, things might look different. This is a really slow system. 16 years seems like a long time for an ecological system, but more time is actually needed. It does seem, though, that an absence of elephants and the presence of all other herbivores is really not good in terms of managing bush encroachment. The riparian areas are quite different, and we need, well, we can't really just generalize these results to the riparian areas. And just end off with a question, the value of these long-term exclosures. It's a really expensive experiment, this one. There was a lot of elephant damage. Eventually, we had to abandon it because elephant damage got too severe. But it's a big experiment, a lot of money and time. Is it justified in terms of understanding how elephants affect Kruger? All right, and I'll end there. Thanks.
Thanks so much, Tony. We're still a little bit ahead. So if there's a question, Ben's got a question for you. Can we?
Thanks, Tony, for a great talk. I was just wondering if you also looked at basal area rather than height for your, if you measured it and if you would see a different picture.
Yeah, so we did measure it. The data's a lot noisier than the height and more difficult to interpret. So it reflects the same trends, but it's not as clear. And it's because of the multi-stemmedness, like new stems coming up, dying on the same individual. Yeah, but we do have the data.
Nikki's also good, so we'll let Nikki. There's another speaker that's actually also not in the session, so we'll take Nikki and then Heidi's questions.
I was just interested to see the actual seedling data. That's really cool. Do you think it's just because of the absence of grass in the control? Or do you think the presence of grass has something to do with it?
Yeah, that's what I would think, but then in the partial, we didn't see so many seedlings, and the grass layer is quite similar. So that's what's really confusing, yeah. It is very patchy, the seedlings. So I do worry that we didn't our spatial replication is not enough. But it is for many years, so eventually that should, you should still see a pattern emerging, yeah. So I'm also wondering if it's not something to do with browser intensities that are slightly different, that we're not picking up with the camera traps.
Thanks, Tony. I also had a question about the seedling recruitment, and I was wondering if in your controls where you've got the herbivores present and inputs from herbivores, dung and urine, that you might have a bit of a faster system there and, more nutrient cycling, and that might be important for the seedlings at their recruitment stage. Any ideas?
Yeah, possibly. Possibly it is. I'm just trying to think if that soil analysis is being done. Not really. I don't think there's anything actually looking at nutrient cycling results. I was also wondering if it couldn't be direct dung effects, like things germinating in elephant dung, except that the majority of seedlings were mapanis, and we know they don't germinate in elephant dung. But also possibly what might be there might be a facilitation effect in terms of branches and stems being pushed over, making little cages that the seedlings can then germinate in as well. And that would also link with soil nutrient cycling, because those sort of, debris piles, if you like, are going to have high nutrients too.
Thanks, Tony. I was just wondering, it does seem like the full exclosure had quite a bit higher densities, and then between 2012 and 2018, it decreased. So don't you think during the drought there was maybe just high mortality in the full exclosure, higher than in the other treatments?
Yeah, so I haven't looked at that. The mortality rates are generally quite low. And there was definitely a lot of dieback, I think in the full exclosure, the dieback rates, the amount of dieback might have been higher. There's just not enough replicates because mortality is so low to actually see if there's a significant difference.
Okay, thanks, Tony. We're going to move on to Karis Corey-Roberts. That's going to tell us about different management practices and habitat used by animals.
Thank you. Good morning, everyone. I'm just going to jump right in since my time is a bit short. So we know that savanna ecosystems exist in a constant flux of vegetation structure. However, climate change and increasing CO2 levels in the atmosphere are leading to a rise in bush encroachment. Globally, savanna ecosystems are managed to control encroachment in a variety of ways, including mechanical clearing and prescribed burning. The effects of these type of management on the vegetation and herbivore communities and herbivore responses to such changes have scarcely been explored. However, understanding these responses are critically important because of their influence on structural heterogeneity, food availability, species composition, and nutritional quality. All of these factors are important to understanding herbivore habitat distribution and habitat selection. The aims of this study were to examine how annual. Most important to note here is the winter mow, which you can see on the far left of the screen in dark blue, showing incredibly low crude protein content and high percentages of acid and neutral detergent fiber. ADF values are inversely related to digestibility, and low NDF values are indicative of more mature forage. So we see that the winter mow is overall lacking in the nutritional components that make for quality foraging. It's also important to keep in mind that this is the nutritional quality for just one grass species that was present across all of our treatments. And moving forward, we'll be looking at species composition of each of the treatments as an additional variable potentially influencing the herbivore response. When we looked at dung counts, we can see the significant differences between treatments was driven by the presence of the herbivores displayed. Note that elephants had the most significant presence on the control and triennial burn treatments, while the presence of wildebeest and impala were most significant for the annual burn and winter mow treatments. Finally, we looked at the density of all dung piles, regardless of species, in each treatment to give us an idea of the overall use. In Kruger, we know that there is good vegetation quality and availability, and that herbivore composition in these treatments is similar. So what we see here is that herbivores actually selected more for the burns than for the control, which we would expect. But when we look at the APNR, note that the use we see in the winter mow is half that of the use that we see in its corresponding control site. Additionally, when we calculated species richness, we also found it to be significantly lower in the winter mow compared to the other treatments. So not only do we see lower density of dung piles indicating lower overall use, but the herbivore community using the winter mow treatment is dominated by wildebeest and impala and is significantly smaller than that in the other treatments. Our results highlight how our management decisions have massive impacts on the ecosystems that we find ourselves responsible for. We see that burning reduces tree cover, but does retain available biomass and quality, having an overall positive impact on the herbivore community. On the other hand, mechanical clearing basically obliterates tree cover and significantly decreases quality and availability of forage, having a negative impact on the herbivores, as we've seen looking at the results. All right, and I'm out of time, but these are our plans for moving forward, so thank you very much, everyone, for listening.
Karis, if you just stay there for a moment, is there a question? We can take one question, Karis, if there's one. Okay. Tony.
All the Kruger sites.
Yeah, so it was very interesting looking at those tree density results. If I can go back to, I don't know if I can go back to the PowerPoint. But when you look at the aerial images, you can see that there's, oh, thank you. Yeah, so it did have overall much lower tree density, and this could be because of the differences in rainfall between the two locations. They're not very, very far apart, but that's why we had a control site associated with each of our treatments so we could compare amongst them.
Okay, thanks, Karis. I just want to point out there are 3 posters connected to this theme that you can look at later today. And now we are going to move on to Ricardo. And Ricardo is going to tell us about how edaphic variation interacts with disturbance to create further patterns.
Hi. All right, so in Savannas, I'm going to go a little bit in a different direction and make an argument for edaphic effects in a tree system rather than a grass system. This is work that my collaborators and I have done in Serengeti National Park. So I think we can all agree that savannas are spatially heterogeneous, and they can change substantially over time. You can see here a massive increase in tree cover in Serengeti over this 23-year period, but you can see that the changes have been unequal. So you've got these open sort of grassy patches, and suddenly they become woodland, but it happens evenly. And so, you know, a question we can ask is, why do some of these grassy patches acquire trees and why do some not? And so we did sort of a multi-year study along transects from high to low tree cover to try to understand which were the demographic processes that led to trees emerging in some areas and not others, and what might the mechanisms be. And in particular, we were trying to understand mechanistically whether these changes were likely to come from below or whether they might be influenced by the trees themselves. That is, do we see evidence of positive feedbacks affecting these changes? Ultimately, we want to understand what is varying spatially in these systems. What we did is we classified and then we identified natural breakpoints that allowed us to separate each of these into areas of relatively high cover and relatively low cover. And we were able to sort of compare a number of things between them. So if we center all of these transects on that transition point, being the kind of relatively sharp transition, tree cover increases quite dramatically over a distance of about 50 meters from the edge of this boundary. So it's very clear that we have areas of high tree density interspersed with areas of low density, and beyond the sort of the shape of this transition, we wanted to understand the mechanisms that might be responsible for generating this pattern in the first place. So one of the things that we did is we went along these transects and we quantified, at ground level, we quantified basal area, seedling density, and the maximum height of trees. And the takeaway message from this is the following. It's very clear that we have clear differences in basal area between the open and the woody sites that match what we see using remote sensing. But we don't see changes in differences in sapling density. It's labeled C. This is that disturbance herbivory prevent these seedlings that accumulate, these saplings, from moving into adulthood. And so we have this bottleneck at around 2 meters height. So what we've seen here is sort of consistent with this previous work. The question is, what is it that really prevents these seedlings from moving across? How do demographic rates of these saplings vary? Things such as their establishment rates, the growth, the levels of damage, the top kill and mortality rates. How does that vary in relation to the tree cover, and what might that be telling us about why they're not recruiting? Which of these processes explains the presence of these bottlenecks? Is it caused by the trees, or is it only correlated with the trees? So we track these seedlings over time. Each of these traces represents an individual sapling. I keep calling them seedlings. The red trace shows the mean for the transect, and you can see that a lot of them are very flat. Kind of looks a bit like Tony's data. Things do not change a lot over time, right? So they're really not recruiting, right? This shows the sort of changes in height over time. You can see in some cases that there's recruitment, but you also see loads and loads of instances of reversal. Top kill, these things get reset back to ground level, they come back again. And we estimate that in the absence of any disturbance, if we take the growth rates during periods when there was no damage, and we projected that the actual mean height of these saplings would be double what it actually was over 3 years. So very clearly there's very strong disturbance going on here, but sort of top-down suppression that's preventing them from recruiting. Now, if we look across, if we look at the differences in those rates between the open and woody sites, we don't see any evidence that there are differences in the rates of disturbance. So for example, fires are just as frequent in the open and woody sites. There is no mean overall differences in herbivore density. We actually find that browsers are more common in the woody sites. So clearly the browsers are attracted to the trees and they are not the cause for the lack of recruitment in the open sites because browsing is less intense there. If we look at the demography of these saplings, we see that the only thing that's related to tree cover is growth. In areas with high tree cover, the saplings tend to grow faster. Disturbance is really intense. If you look at the bottom panels show top kill and damage, the rates are really high. Those are annual rates, but they're flat as you move across from open to woody. So there's no sort of clear pattern there between tree cover and disturbance, even though disturbance is tremendously high. And grass biomass doesn't really show a clear relationship either. It might show a slight positive trend, suggesting that whatever is promoting trees is also promoting grasses. So, growth is the only thing that really sort of varies across this gradient. And so what we did is we wanted to understand how would this kind of play out over the long term, right? Because we have 3 years of data. These are sort of really slow rates. If we take them and try to project them forward using a simple model, what would they tell us? And so what I'm showing you here are the results of a simulation that's fed exclusively from the data that we collected in the field that projects the probability of escape across a gradient from no trees to 40% tree cover, based on purely on these sort of correlative relationships between demography and tree cover. And the case in the left takes our data at face value. It sees sort of a relationship between growth and tree cover, but no relationship with disturbance. And the scenario on the right assumes that growth is just flat, but that top kill and damage may vary, even though the trend was not significant as a function of tree cover. And what you see is that the case on the left predicts that there is a dramatic increase in the probability of escape in areas of high tree cover compared to the open areas. So there is almost no escape in the open areas, which is consistent with the fact that we don't see a lot of that recruitment and the presence of that strong demographic bottleneck there as opposed to the areas with higher tree cover. If we assume that what varies spatially is disturbance, we see the results on the right. It shows that nothing should ever escape. Now, it's not that disturbance isn't important. It is tremendously important. It works together with growth to give these kind of projected patterns of escape, and these projected patterns of escape may therefore explain the patterns of tree cover that we see. If we theoretically relax disturbance in the system, the patterns start to shift a little bit and eventually disappear, suggesting that that strong disturbance is important, it's just not spatially varying. So then a question is, we have a correlation between sapling demography and tree cover, but what's driving it? Are the trees themselves driving it? This is where our sensor data comes in, becomes helpful. Do we see evidence of sort of microclimatic effects of the trees that may be influencing some of these rates? Is tree cover the cause of variation in growth or simply a manifestation of it, an outcome of it? I think the second answer is the correct one. So if we look at the sort of soil moisture microclimate data, what we find is that we have plenty of light availability in both areas of high and low tree cover. We have actually slightly lower soil moisture in areas of higher tree cover. So soil moisture is clearly not limiting there. And we don't see really strong patterns with temperature, suggesting that microclimate and sort of ameliorative effects of tree canopies on the growing environment for the seedlings are not really sort of explaining the patterns that we see. So we don't see, based on the microclimate data, we don't see very strong evidence of facilitation occurring. What we do see is very clear differences between the soils. We made water retention curves for soils from open areas and woody areas, and in all of these cases, the line to the right is the line for the open habitats, and the line to the left is the line for woody habitats. And what you have on the y-axis is pressure potential, and on the x-axis we have the soil moisture volumetric content. And what it shows is that for any given volumetric content, conditions are much more stressful in the open habitats than the woody habitats. So that suggests that we have stressful soil conditions that may be limiting growth, and that just subtle difference in growth may be responsible, may prevent those saplings from ever escaping that very strong disturbance trap. So to summarize then, we have these very kind of strong spatial transitions in tree cover in Serengeti, and we believe that they can be explained by discontinuities in the probability of escape, and the discontinuities themselves are then explained by a combination of a subtle growth gradient that may be caused by moisture stress, coupled with a very intense disturbance regime. And so one of the conclusions is that the bottom-up drivers have a sort of spatial signature, the things that control growth, such as water stress, whereas disturbance, while being incredibly important, does not show a strong spatial pattern. So one possibility that we could entertain would have been this one, that the tree is moderating disturbance effects and promoting saplings. The other, and this is not really supported, the other possibility is that there is an underlying edaphic gradient that is promoting higher growth in saplings in areas, in some areas, and then those eventually become the adult trees. And we think that this is the better explanation. Thank you.
Thanks, Ricardo. I was so lavish in my question time that I think we now fallen behind, but Sam say we can take one. Is there a question? Sorry, Ben, Lawrence beat you with like a second.
Yeah, so I'm saying that we don't see clear evidence in this system that the microclimate is having much of an effect. What we're saying is that we think that the adult tree patterns are ultimately just kind of cascading out from the patterns that we see in the saplings. Yes, so because those tend to be like, some of those areas have, they have more clays, and they're kind of, it's more stressful for trees to be able to extract that water and transpire and grow, maybe explaining the lower growth rate of the trees in those systems. So those trees sort of become more vulnerable to repeated disturbance because they never get to the size where they can finally escape disturbance. And so they're perennially trapped in that first kind of lump in that distribution that I showed you because of those low growth rates. So I mean, a lot of people have shown that these systems are very sensitive to growth, and I think this sort of adds to that variation in growth.
Hold it before you run. Quick question. Did you look at grass composition and biomass differences between the sites? Could it be that under the trees you got a different community of grasses with less competition, and outside more grass, more competition for water, which could explain the drier soils?
So there's definitely compositional patterns, yeah. Just like you see in Kruger, you see more Thermida in the open areas and more Panicum maximum under the trees. So I don't know, but it is interesting that as the grass biomass tends to increase in the areas of higher tree cover, the tree growth rate also increases there. Which is not to say that grass competition isn't important. I think it's hugely important. I think what I'm arguing is that its intensity doesn't vary tremendously across this gradient. But I can't completely rule out what you propose here.
Okay, thanks so much, Ricardo. So now we're going to move on to the last talk of this theme, and then we're going to get back to Kate. And I just want to point out that there's one poster also for this theme. Fabio couldn't make it, but he sent Luca Malatesta. Thanks, Luca.
Thanks, Corley. So good morning, everybody. Today I'm going to present you the first results of this study in which we are trying to understand the relationships between elephant covers and tree density at the scale of the whole park, repositories. So before observed a positive correlation of tree cover with temperature seasonality. On basalt, on the other hand, we have the opposite situation in which elephant bulls that were not significant in affecting tree cover on granite due to their scarce density. Elephant bulls negatively affect tree cover or are negatively correlated with tree cover on basalt, while elephant herds have a positive correlation with tree cover on this substrate. Tree cover is also positively correlated with sand component in soil. So what we observed from all this data are some take-home messages that you have here, and these are take-home messages also for us, for our future homework. The main factor that will influence also our future analysis is that the geological substrate sharply differentiates elephant density, in particular bull's density, and tree cover, and the interactions between the two. On basalt, we have seen that bulls reduce tree cover and density, and this is probably due to browsing and felling of trees. So what we observed is probably these causal relationships in which bulls reduce tree cover, while the positive correlation of herds with tree cover and density may probably be due to the opposite effect, in which herds prefer favorable environments that are that have a higher tree cover and density, given that on basalt, in general, tree cover and density are very low. On granite, we observe something that may point in the direction of a mutual influence. So what we have to do in the future to understand better these relationships at the scale of the whole park is a multi-temporal analysis with these three separate tasks, which we have already started. So we will analyze tree cover changes from the 40s, elephant population dynamics, and also relate all this with fire frequency and intensity history. The tree cover analysis has already been started. We already have the first results, and those will be presented by my colleague Tulia in the next days. Thank you. Thank you very much for your attention.
Thanks so much, Luca. I think we're going to move to our last speaker before tea. Kate, sorry for making you wait. And Kate is going to tell us about
great talk about the Armageddon experiment. Conversely, so you mentioned the potential impact, but I mean, a lot of areas are also experiencing huge impacts of invasive ant species, like the big-headed ant. What could your results potentially predict about that?
Yeah, so if there are invasive ants that are having a particularly strong effect, they may reduce termite abundances. But my experience is that quite often those invasive ants are just replacing what the native ants are doing in terms of predation, I think. And in our system here, there is the big-headed ant, Phydolia megacephala. It's a native ant, and that is one of the key predators. It's a tiny little ant, you know, you don't give them much notice. Everyone thinks it's the big ants that are the big, scary predators, but actually these really tiny ones can also do a lot of damage by the looks of it, to termites, if you're a termite. Okay.
Thanks, Kate. Corley's left me, so I'd like to thank all the speakers in this session before I carry on with my announcements. So just a couple things from my side. Guys, just to remind you, the water bottles, you only get one a day, so if you are leaving the session and you've been sitting at a bottle, please just take it with it and bring it back when you come back. Then if anyone doesn't want their talk streamed, please tell us in advance, because it's a little bit of work for the guys here to set it up, so we're going to then move you to the end of that session. So we'd just like to know that in advance, please. Then also the meal tickets, please remember to take your meal tickets with you at night time. Tonight at the golf club, they are actually going to be asking that, so you won't get a plate if you don't have a meal ticket, so please just do that for us. And then lastly, the bags and the lanyards this year have been made by a local community sewing group. You'll see there are some pamphlets outside advertising that. We do have a few spares, so we are selling those. So if you're interested in buying a bag for somebody back home, please then just check to Jackie and get one of those. So we are a bit late, so I think let's start 5 minutes after the starting time after tea, so that'll be 11:05. Thank you.
Okay, welcome back, everyone. In the interests of getting you to lunch on time, I'm going to get us going in this next session. Before we do that, I also just like to draw your attention to the screen here. Kathy has asked us just to let you know that they need assistance with everything will be shown up on this thing. It'll be playing during breaks, and so please just do have a look. And if you are able to help, please contact Kathy. All right, so I'm going to get going. I know there's still people missing, but let's start the session. First up, we have Clara, and she's going to be talking about the relationship between browsing pressure and species shrub richness in Kruger National Park. Thank you.
Okay. Good morning, everyone. It's really nice to be back here. My name is Klára Pišková. I'm from the Czech Republic, and I'm a PhD student at the Faculty of Science at Charles University in Prague and the Institute of Botany, and I will be talking about browsers and shrubs. Honza Čuda, our colleague, he gave a talk this morning about grazers and grasses, so I'm going to give you basically the same presentation with different, closer? Okay, with different animals and plants. So browsers are quite a diverse group of herbivores, not only in terms of their ecologies, but also their body sizes. We have them spanning from the tiny steenbok all the way up to the biggest land mammals, elephants, giraffes, and so on. There are 16 browsing and mixed feeding species in Kruger National Park, but some of them are strictly browsing, have a strictly browsing strategy. Others switch their diets according to seasonal availability or palatability of the vegetation. And I'm just going to be calling all of them browsers because I'm talking about browsers as a functional group. They are more influenced by plant mechanical defenses than grazers because shrubs, they have more defenses either in forms of some mechanical, like thorns and stuff, or they also provide much more diverse and toxic chemical compounds that the browsers are affected by, so maybe they, because of that, they choose more what they want to eat. So obviously the distribution of browsers is affected by many factors, but vegetation is definitely one of them. And then in turn, browsers can definitely affect the vegetation that they feed on significantly, as we heard many times. And we wanted to have a closer look at the relationship between the browsers in Kruger in different habitats and the effect they have on shrub cover and shrub species richness. So it's a part of the Mosaic project that Honza already mentioned in his presentation. So our project focuses on biodiversity and identifying some patterns and interaction between animals and plants and looking at some environmental factors. And our hypothesis for the Mosaic project was that seasonal rivers are an important factor in maintaining biodiversity in Kruger or in a savanna. The reason for that is that as you get closer to water, to river, you have an increased vegetation productivity, but at the same time you have a much higher disturbance pressure from the large herbivores. With seasonal rivers, the situation is a little different because in the dry season you don't have any water in the rivers, so the vegetation productivity is decreased, but at the same time the disturbance pressure is also decreased. So at these sort of intermediate levels of disturbances and stress from the drought, we expect this might yield to some higher diversity of vegetation of some species, or perhaps presence of some more rare species or species that are less resilient to these disturbances by the herbivores. And that could affect the diversity of other taxonomy groups. So this was already mentioned. We have 3 different habitats which are placed on a gradient of water availability. So we have plots at perennial rivers, seasonal rivers, and then dry crests, and we sample these taxonomy groups. And I will be talking about plants and mammals today, but we also will have some more talks about birds and bats later during this conference. So I was mentioning some environmental factors. So when we were selecting the plots, we were also considering bedrock. So as we all know, we have basalt and granite soils in Kruger. We were using the 4 main land systems, bedrock combined with the dominant vegetation type structure. Obviously, I said we were monitoring water, rivers, so that, and the crests were selected in areas that are at least 5 kilometers away from water or any water source, so the yellow or red areas. We ended up with 15 plots in each of the land systems. They were scattered nicely across the whole park. And we ended up with 20 triplets because each triplet has always one site with each habitat. So 60 plots in total, and in each of those plots we collected data on all these taxonomy groups. So for the botanical data, first they were collected by our botanists, Honza, Martin, and Petra, and they were collected in all of those plots, 50 by 50 meters. They did a complete botanical survey. They wrote down all the species they found in the plots and also recorded their abundances expressed as percentage cover. It was done in the rainy season of 2019 and 2020, and this is kind of a background for the rest of the talk. So this is data on shrub richness and shrub cover. So we didn't find any significant differences in the shrub cover among the different habitat types. That's the graph on the right. And then, And then the shrub species richness was significantly higher at seasonal plots, which is this bar. And for the herbivore data, they were collected with camera traps. We had 1 camera trap in each of the plots, so 60 of them in total. For this talk, I'm going to be using the term abundance, but actually it's just a total number of records of the herbivores, of the browsers, because I take it kind of as a proxy of habitat utilization. So if the animals decide to stay in some habitat, some plot for 3 hours, rather than just quickly pass through, it probably for some reason is more suitable for them. Or maybe they feel safer, have more food, whatever. The data are from both rainy and dry season, and we were focusing on hoofed mammals of the browsing guild. These are the species that we recorded. We didn't analyze the data for sable and cliffspringer because we only had a few records. So that was 10 species, I think, we used for the analysis. This is a total number of records for all of the browsers. So yellow color is in habitat. The habitat was seasonal river. Blue is perennial river, and then the red one, the red part is the crests. So shockingly, impala was the most common herbivore. And if we remove impala, then of course elephant, and then the rest of the species were present much less. This is just a sum of all the records of all the browsers in the different habitats. So crests had the lowest number of records of browsers, and perennial rivers had the highest number of visits. So now on to the data, the analysis. So in the beginning I mentioned shrub cover and the relationship of browsers with shrub cover. There wasn't any relationship, so that's the first question answered. So I'm just going to focus on shrub richness, species richness. So we did find a marginally significant effect of the interaction of browser abundance and habitat on shrub species richness, and then a significant effect of species richness of the browsers. So the first one, the interaction. So in this case, that's the marginally significant interaction between browser abundances and habitat on the shrub richness, species richness. I'm showing a graph for each of those habitats separately. For crests and seasonal rivers, we can see a strong effect a strong relationship, but we don't have any relationship for the perennial rivers. So I have a couple theories why this might be. So the shrub species richness is increasing with the browser abundances at crests and at seasonal rivers. So either this could be a pressure from the browsers, and they might work as kind of a dominance control at the crests and seasonal rivers. The more animals, the more they might suppress some more common species of the shrubs, and that could potentially be dominant. Or it could be the other way around, that the more shrub species there are, the more attractive that habitat is for the browsers, because they will have a more diverse buffet. They will have a higher chance that they will find the species that they prefer. And the perennial rivers, perhaps the disturbances are too strong, because this is just a reminder that the perennial rivers had the highest traffic of herbivores. So maybe the disturbances are too strong for some species to survive. And then this is the second relationship that was significant. So that's browser species richness and shrub species richness. So the species richness of shrubs increases to a certain level. Oh, and I'm only showing one graph because there wasn't any interaction that was significant with habitat. And you can see the different colors in the graphs represent different habitats. And indeed, the relationship is not structured by habitat. So up to a certain point, the shrub species richness increases with the browser species richness, and then it goes down. So maybe it is because the more browser species there are, the higher the chance that some more specialized species will be present that will potentially control some potentially dominant shrub, kind of like it was with the abundances of the browsers. But then when the browser species richness is too high, then there's a higher chance that there will be more herbivore species feeding on a particular species, and that might suppress or eliminate it. There was a correlation. It was significant. It wasn't very strong, but it was there, between abundances and species richness of the herbivores, of the browsers. So perhaps we also, with higher species richness, we also get a higher browsing pressures from the herbivores. So the next step would be to have a look at some traits of the shrubs, like palatability, or because, as I mentioned in the beginning, they can produce quite a lot of defense chemicals, which influences, of course, what the browsers pick. But also having a closer look at the specific preferences, feeding preferences of the species of browsers. So yeah, those are my hypotheses, but we still have to look more into it. This is quite preliminary. So just to sum it up, to conclude, the species richness of shrubs is highest at the seasonal rivers, and the shrub cover didn't differ among the different habitats. The abundances of browsers was highest at perennial rivers. We didn't find any relationship between the browsing pressure and the shrub cover, but we did find an effect on the species richness of the shrubs. And the effect of the abundances of herbivores of the browsers was most significant at crests and at seasonal rivers, but there wasn't any relationship at the perennial rivers. And the species richness of shrubs is highest at medium browser species richness. And that's it from me. I want to thank our game guards for keeping us alive, and all of you for your attention, and everybody else who helped. Thanks.
Okay, thanks, Clara, for a great talk. We do actually have a couple of minutes for any questions. So please, anyone got a question? Yes, Tony.
Hi, thanks. I was just wondering, about forb abundances and diversity? Because, I mean, a lot of those browsers are relying a lot on forbs in the herbaceous layer. So do you have that data? Can you take that into account in trying to understand the relationship with shrubs?
I'm sorry, can you repeat it? I can't.
I was asking about forb species.
Yes.
Yeah, so can you work that into your analysis? I mean, I'd imagine that plays a big role in where the browsers are or are feeding.
I'm sorry, I'm having a trouble understanding. Actually.
So do you have data? Do you have data on the forb species abundances?
Yes, that's
Yeah.
the shrub, the cover.
The cover, yeah.
Yes, it didn't differ among the habitats.
So I was just wondering why you haven't looked at that in your relationship, because that must have a big effect on your browser abundances.
We didn't find any relationship between the browsers and the shrubs, the shrub cover.
Okay, any other questions? Okay. Francis?
Yes, thank you for a very interesting talk, and also yes, to just follow up with Tony's question about the forbs. I also think that we find it very difficult to distinguish between a shrub and a forb in these systems, and I'm just curious to now know whether you have, how did you decide whether it's a shrub or a shrub species, or when it's a perennial forb that's very shrub-like?
So I am not a botanist. I am in charge of the animal part, but I think they were they were like looking at the layers of the vegetation. So one species could be in the herb layer as well as shrub layer. So it was more about the life form than some, but I'm not sure. Sorry. Maybe Honza there behind you can answer.
Okay, we have run out of time. Yes? Okay, great. Clarice is going to do a 5-minute talk on interactions between foraging hotspots and browsers. Thank you, Clarice.
Greetings, colleagues. Have you ever wondered if foraging hotspots defined to a key browse species facilitate interaction between livestock and wildlife? If you have, well, today I'm going to give you the answer to that from research done at Great Zimbabwe University and the University of Zimbabwe, sponsored by the Research Platform Production and Conservation in partnership. My name is Clarice. Please move. Okay, if you haven't wondered, then you might be wondering why the question. For people living at the periphery of conservation areas, and also from studies that have been documented, it has been shown that proximity to conservation areas actually is characterized by conflict that includes zoonotic disease transmission, as well as competition between livestock and wildlife for feed. Work done has shown that there is animal movement at the interface in terms of resource gradients between conservation areas and agricultural areas. Agantua et al. In 2011 showed approach grazing, whereby farmers were shown to drive the animals as deep as 10 kilometers into the park to look for areas where there was grazing during the dry season. Now, these areas where there is livestock or foraging during the dry season are what we are referring to as dry season livestock foraging hotspots in this study. Even though we have those studies that have been documented, there is limited empirical evidence on the geographical location of these foraging hotspots, as well as what they really look like and whether they facilitate interaction between livestock and wildlife. So the moment you're able to answer that question, we're also able to enhance coexistence at our interface, because these are considered some of the drivers of livestock-wildlife interactions. So the objectives of this study were to detect dry season livestock foraging hotspots, explain their link with key browse species, as well as determine whether location of dry season foraging hotspots influences interaction with wildlife at the interface. What we did was, our study area was the southeast lobe of Zimbabwe, studying communities at the periphery of Kruger National Park. So the first thing we did was to produce probability distribution maps of key browse species, then detect these foraging hotspots using kernel density estimation and nearest neighbor hierarchical clustering, as well as to look for association between livestock and wildlife using multi-distance spatial cluster analysis. For our results, that's what we came out with. That's a map of combined probability distribution of key browse species. So we're looking at colophos, Sperma mopane, Dicrosarchus cinerea, and Salvadora perstica. So from that map, you can see we had stands of Dicrosarchus cinerea on its own. We had combined mixed woodlands of the two species, none of the three. Then we had 3 foraging hotspots that we detected, and there was high overlap between the two methods, showing that we had confidence that what we had detected were actually the foraging hotspots. And then looking at the composition of the hotspots, we found out that we had Dicrosarchus cinerea is the most common species in the foraging hotspots. We also had mixed woodlands of Dicrosarchus cinerea and mopane. And that's not surprising because Dicrosarchus cinerea actually is very abundant and persistent during the dry season. Then also had, looking at the frequency of sightings of elephants, I forgot to mention that elephants were used as a proxy for wildlife in the study. And then we also had livestock movement data that was being used. So we had 2 peak sightings, one inside the hotspots and one that was influenced by water, showing that water also influences occurrence of foraging hotspots. And then average elephant sightings from the park boundary with 0.279 kilometers. But although we had only 0.003% of these sightings inside the hotspots, 59.5 of these were within 5 kilometers of the hotspots and 68.5 were on the park boundary. And then we also showed that there was a significant clustering between elephants and livestock using the multi-distance spatial cluster analysis. And in conclusion, from our results, we suggest that key browse species are concentrated around browsing hotspots, and thus their location could facilitate interaction between livestock and wildlife. But knowing that there is a resource shift in resources, we recommend looking at what would be the foraging hotspots at the beginning of just before the dry season begins. And also further studies could look at foraging behavior of other wild herbivores on larger landscapes, so that we can really have a better conclusion of whether or not we have these interactions. And lastly, we also want, this is important because of implications of stocking rates, rangeland carrying capacity, and other management and conservation strategies. You can have a look at some of those papers for some of the data I was talking about, and thank you very much to everyone shown on the slide. Thank you.
Okay, thanks, Clarice. Unfortunately, your 5 minutes are up, so we will move on. Next up, we have Philippine, and she's going to be talking to us about using DNA metabarcoding to look at diet overlap between black rhino and other browsers. Thank you.
So, good morning, everyone. Today, I'm going to present to you my master thesis, which is on dietary DNA metabarcoding, a niche overlap of the black rhino and other browsers in South African reserve. So as you all know, the black rhino is a critically endangered species, and a big part of the population is living in closed area. So they are being intensively managed and along other species, and in order to have optimal management, one needs to understand ecological interactions between species. So an ecological niche is defined by the fact that a species needs environmental conditions that permits it to thrive, and that biotic factors such as predation and competition impact the species niche. Diet is an important component of a species niche, and so it's important. Analyzing diet gives important information on community ecology and also on the possible existence of competition. So in our study, we aim to investigate the extent of diet overlap between the black rhino and other browsers, And because other browsers may also impact black rhino through their density and also their browsing pressure. So this study was conducted in Itala Game Reserve, KwaZulu-Natal. As black rhino is a browser, any species that also feed on the same vegetation types can encroach on black rhino's dietary niche. So the greater kudu is a strict browsers, whereas elephants and impalas are mixed feeders, so they also feed on grass. So as you can see, our data collection consisted in collecting all shapes of fresh dungs all over the reserve. So we collected the samples in the preferred areas of the black rhinos, and also in all quadrats of the reserve, as well of the main vegetation types. So we use DNA metabarcoding, which is a non-invasive and accurate tool to analyze the herbivores' diet. So after having collected the fecal samples, we extract DNA, and then we amplify a DNA marker, and we selected the RBCL plant DNA marker, which is a standard one, and also it's a short one, because DNA has gone through a whole digestive system. It's highly degraded, so it's important to choose a very short DNA barcode. Then we sequenced it with the Illumina technology, and with the help of bioinformatic analysis, we could assign taxonomically plants to species. We also did some statistical analysis. We we looked at species richness, diversity, and evenness, and we also calculated the Bray-Curtis dissimilarity index, as well as the Pianka's niche overlap index. So regarding our results, we successfully amplified 94% of our samples, and we were very happy about that. And we also identified 300 genetically distinct plants in the herbivore's diet. Because DNA metabarcoding is, because sometimes the barcode was not distinct within a family, we could identify 31% to the species level, 71% to the genus, and 96% to the family. So here are some pie charts illustrating the herbivore's diet. So we can see the 10 most common plant families. So we analyze the herbivore's diet in general. So I'm not going to go into details, but we can see that they had significantly different diets, and also that diets changed with the season. Regarding the species richness, so the herbivores, they fed on more plant species in the wet season, and also the black rhino, they ate very less plant species than other herbivores, whereas the kudu ate on average 29.4 plant species per sample. Now the species evenness. So the species evenness is the relative abundance of each plant species in the herbivore's diet, meaning that if the evenness is low, some species are more eaten, and when it's high, the plant species are more even in the herbivore's diet. So in general, the evenness was lower in the dry season, so they preferred some plants when the choice was extensive. And the black rhino had the lowest evenness, showing a higher preference for some plant species, whereas the elephant had the highest evenness. Now, regarding the species diversity, we calculated the Shannon Diversity Index, which is calculated with the richness and evenness. So we can see that the diversity was higher during the dry season, And also, again, the black rhino had the lowest diversity in general, and the kudu had the highest diversity, not far from the elephants and impalas. Then regarding the dietary overlap, so we used the Bray-Curtis index as well as Pianka's niche overlap, and we can see that in general the overlap was high. It was of 82%, and that impalas encroached the most on black rhino's diet, which was quite surprising, followed by elephants and then kudus. Now, when we look separately at season, so we can see that in the wet season, the overlap was of 84%, and in the dry season, the overlap was of 65%. So there is a drop of overlap of 20% from the wet season to the dry season, which is also quite a big difference. And then, so this is a river plot illustrating the most consumed species by the 4 herbivores. So 30%, almost 30% of the herbivores eaten plants was Dicrostachys cinerea, belonging to the Fabaceae family. And so we can see that they ate a lot of similar plant species, and the overlap was high on those ones. So to summarize, the black rhino is confirmed to be a selective feeder with our result, and they had the narrowest dietary niche. Kudu, they had the widest dietary niche, and our result revealed that in Itala, they tend to be more, if I can say, generalist browsers. Now, concerning impalas and elephants, so they are mixed feeders, so it was expected that the dietary niche would be wide, and that's what our results revealed. Then we can see that the diet shifted with the season, and that when resources are abundant, herbivores forage on more plant species with wider variation in abundance and less diversity. So this means that when their choice is extensive, they have more preference for some plant species. And then we also, with our result, we could see that the overlap was high in the diet of these 4 species, and that when food is abundant, the overlap was higher, whereas when the food was short, it was lower. So interspecific competition promotes the use of different resources. So a shift in diet can be correlated with the intensity of competition, and herbivores compete more for resources in the dry seasons, which makes sense. Then regarding the overlap intensity, impalas, they encroach the most on black rhino's diet, but when we take into account the body size and the forage intake, they probably don't have a lot of impact on black rhinos, or even though they eat more the same plant species than elephants. So they eat on average 1 kilo of forage, whereas elephants eat 170 kilos. So the elephants probably have a higher impact on black rhinos. And also our results were quite surprising, because we were expecting that kudus might encroach more on black rhinos' diet as they are browsers, but in Itala it was more the impalas. And then with regards to resource abundance, so some plant species were very common in the herbivore's diet, and this is probably linked to the fact that they are widespread and common in the reserve, and that the herbivores encounter all the time these plant species. So these common species outweigh rare species in the analysis, and it's important to also point that out. So in conclusion, we could see that there is a large dietary niche overlap between these four browsers, and that interspecific competition drives resource partitioning. So such processes are more apparent when resources are limited. And this mechanism allows species to coexist by having more distinct niche in the dry season. We also see that DNA metabarcoding is a great conservation study tools that gave important information on species assemblage and herbivores communities, and also gives important ecological knowledge on a critically endangered species. So the further steps would be first to do vegetation surveys on plants resources abundance, and to build an electivity index to give more weight to rare species, as they could be more limited for herbivores. And also one limitation of DNA metabarcoding is the lack of distinction between plant tissues. So some herbivores might eat, well, they might eat the same plant species but not the same parts. So some herbivores might eat the leaves, whereas some eat the bark and other roots. So that would be interesting also to investigate, as well as feeding height and secondary metabolites. And finally, it would be also interesting to take into account habitat use overlap in further studies. So thank you very much for your attention.
Thanks very much, Philippine, for a great talk. We do actually have time for a couple of questions. We've got one over there. Do we have another mic?
Thanks, that was a nice talk. I had a question about the DNA metabarcoding approach. Can you weight the amount of forage consumed by species so it more accurately fills out the complete diet? Because a species that's quite rare, that it was only a portion of one leaf was consumed, you wouldn't want to treat that the same as a species that comprises 90% of the total forage intake. Can you use this approach to separate that out?
So you can actually see the relative abundance of each sequence in a sample. And we haven't done that, but it's certainly possible to know the quantity of each plant species in a sample.
Yeah, thanks very much. That was fantastic. I'm thinking of the dry season stuff that you highlighted, and the one thing you say is that we see this split, and it's actually quite exciting because the black rhino are feeding on one source and everyone else is shifting away. And you suggest competition, and I don't know if I actually buy that, because what you may have is a situation where those species can't handle this to maintain on those same species. They have to shift, they have to broaden their diet breadth. Mm-hmm. It isn't a competition that black rhino is shoving them out, it's the fact that they can no longer handle those species where the black rhino can. So they continue to feed, have a lower competition in that dry season, which is the critical time for herbivores. So I would maybe think about that a little bit to maybe get away from the competition and see more of it as black rhinos being able to maintain and not have to shift.
Yeah, that's really interesting and a good point. And we also see that, the diets, in the wet season, they all tend to eat the same resources because it's abundant and they can eat what they prefer. Whereas in the dry season, the niche separates, which is really nice to see. And probably also it's interesting to see which actually species black rhinos can actually eat, whereas other species can. But thank you for the comment.
Susie, was it you trying to go incognito in the moving around? Last question from Susie, and we'll move on.
So we didn't take into account giraffe because they obviously feed on the canopy of the trees.
Okay, great. Thanks very much. Next up, we have Jyoti. Another speed presentation, 5 minutes. Thank you.
day, everyone. Today I'll be presenting a chapter of my master's thesis. Black rhino was the most abundant of the world's rhino species throughout most of the 20th century. Due to large-scale poaching between 1970 and 1980 caused a 98% decline in the rhino population numbers. To ensure their continued existence, they are now restricted to protected areas. The specific objective of my study was to determine the seasonal, diet preference, and browse availability of introduced black rhino species in Welgevonden Game Reserve. So my study was based in Welgevonden Game Reserve, which is a reserve in Limpopo province of South Africa. The reserve is situated in the Waterberg Plateau of the Waterberg mountain range. Welgevonden introduced black rhino in 2018, which was one of their drought periods. And this raised a question to management as to if there was sufficient forage to support a viable population of black rhino in the reserve. So I conducted fieldwork on a seasonal basis, first in the dry season, which was June, July of 2020, in the early wet season of November, December 2020, and the late wet season, which was March and April 2021. This was to show the seasonal preference for the black rhino. So for the diet component, I did histology, a microfecal histological analysis, and this method provides an estimate of the ingested biomass per plant taxon in fecal sample. So leaves were collected in field and brought back to the lab where they were dried at 70 degrees for 48 hours. They were then boiled in nitric acid for 5 to 10 minutes, and then this layer was removed and permanently mounted in a microscope slide and stained with hematoxylin. Under the microscope, I photographed them to create a plant reference collection. Dung collected from the characteristic black rhino middens were collected and brought back to the lab where they were dried at 60 degrees for 72 hours. 50 grams of the dung was boiled in 20 moles of nitric acid for approximately 5 minutes. They were then spread homogeneously over a microscope slide under 2 cover slips. To assess the browse availability, I conducted sampling plots, and for trees, I obtained 2 very important measurements, which was the canopy cover, and this is the area covered by a plant's edible matter, and the canopy depth, which was the vertical distance between the highest and lowest point of edible browse, which was at 2 meters. Previous studies conducted showed that black rhinos are browsers with a broad diet. However, they focus on certain plant species which make up bulk of their diet. This was shown in Welgevonden as well, where 38 plant species were identified as potential browse for them, but however, the histology showed that only 26 plants were eaten. 23 species were represented in the dry season, 20 in early wet, and all 26 in the late wet season. Plant species observations in the dung from the histology showed that of 3,984 observations in the dry season, Euclea natalensis dominated the diet. From 1,479 observations, Sisyphus mucronata dominated the diet. Observations, Ocna pulchra dominated the diet. They also showed a shift from evergreen species in the dry season to deciduous tree species in the early wet and late wet season. So with fieldwork conducted in the transitional period between the dry, early wet, and late wet season, overall patterns of dietary selection by black rhino and Welgevonden Game Reserve showed clear indications of seasonal change in plant species preference. Even though their diet was diverse, only a few made up bulk of their total forage intake. So assessing the overall availability of browse species for new populations of black rhino are vital for calculations of ecological carrying capacity. Not only this, but also to conduct habitat suitability in conjunction for the reserve. Thank you.
Thanks very much, Jyoti. You are out of time. So we will moving on to elephants and the role of monoterpene concentrations in determining their diet. Thanks, Tuane.
All right. In a previous study, we looked at the influence that individual monoterpenes have on elephant diet choice at a fixed high concentration and found that most monoterpenes deter elephant consumption. The question that remained, however, was whether this deterrence would diminish at lower concentrations. Like tannins and nicotine, monoterpenes are plant secondary metabolites that can act as toxins by damaging cells or disrupting cell processes, or as non-toxic dietary inhibitors, which decrease the nutritional value of food. Herbivores can cope with the negative effects of plant secondary metabolites via physiological mechanisms such as detoxification enzymes or behavioral mechanisms such as avoiding or limiting the ingestion of certain foods. In order to implement these behavioral mechanisms, some herbivores learn to associate the post-ingestive feedback of consuming a plant with the pre-ingestive cues of the plant itself. Volatile organic compounds like monoterpenes, for example, emit strong, distinct odors and can provide a pre-ingestive olfactory cue while also incurring post-ingestive costs. The African elephant is an excellent example of a mammalian herbivore that uses pre-ingestive olfactory cues. In particular, elephants tend to avoid woody plants with high concentrations of monoterpenes. However, certain monoterpenes are more abundant in avoided woody plants than others, which implies that elephants have different tolerances towards these compounds. This brings us back to the question, does the deterrence of an individual monoterpene change with concentration? To answer this question, we traveled to the Adventures with Elephants facility in Bela Bela between the months of August and November in 2019 and 2020. There, we ran odor-based choice trials on 5 semi-tame adult African elephants to determine elephant diet choice between foods without the added odor of a monoterpene and foods with the added odor of an individual monoterpene at a certain concentration. During these trials, we presented the elephants with a choice between 2 buckets. Both buckets contained a sample of Euclea crispa as a food reward, which is a principal plant which is frequently encountered and consumed by the elephants. The buckets were both covered with perforated lids, which allowed the elephants to smell but not see the contents. Along with the Euclea crispa sample, both buckets also contained a 2 milliliter Eppendorf tubule taped to the inside near the bottom. This tubule either contained dipropylene glycol, a largely odorless solvent, as the control, or an individual monoterpene suspended in dipropylene glycol at a certain concentration. In total, we tested 8 monoterpenes at 5, 10, 20, and 30% concentration. The total amount of monoterpenes within these solutions ranged from being 1% of the dry weight of the food sample to 10%. Similar values have been used in previous studies done on brush-tailed possums and sheep to determine the influence that individual monoterpenes at different concentrations have on herbivore diet choice. In these studies, 1% dry weight was considered to be a very low added level and 10% a very high added level. The trial started off with the elephant a few meters away facing the opposite direction. It was then instructed to turn around, walk up, and smell each of the buckets. After smelling both buckets, it was instructed to indicate its preferred choice by placing its lid on the chosen bucket. The bucket that was not chosen was taken away, while the chosen bucket's lid was removed to allow the elephant access to the food sample. As previously mentioned, the monoterpenes used in this study has different abundances in avoided and preferred plants. Linalool and gamma-terpenine are both more abundant in preferred plants. But as we go down this list, you can see that the abundances in avoided plants increases considerably, with terpineline roughly 2,000% more abundant in avoided plants. As such, we expected the elephants to be more tolerant towards the monoterpenes at the top of this list, and that these monoterpenes would likely not drive elephant avoidance across all concentrations. In contrast, we expected the elephants to be less tolerant towards the monoterpenes at the bottom of this list, and that the monoterpenes would be more likely to drive avoidance across all concentrations. Each elephant was tested 5 times per monoterpene per concentration. As such, we used the data that we gathered as repeated measures in generalized estimating equations, or GEEs, which compensate for the possible lack of independence generated by repeated measures. The results of the GEEs were back-transformed for graphical representation, like the graph shown here. The dots indicate the mean proportion of elephants avoiding the monoterpene. Random selection occurred where the 95% confidence intervals overlap with the 50% proportion line, like at 5 and 10% for linalool. In contrast, the elephants avoided the monoterpene when the 95% confidence intervals didn't overlap with the 50% proportion line, like at 20 and 30%. Both linalool and sabanine were at the upper part of the list previously shown and were not avoided at lower concentrations. This implies that the elephants can indeed tolerate these compounds in low quantities. However, despite being more abundant in preferred plants, gamma-terpenine was avoided across all concentrations. A possible explanation for this might be that Euclea crispa already contains high levels of gamma-terpenine. So, in reality, gamma-terpenine was being tested at far higher concentrations than the other monoterpenes. It is likely that if we were to use a different food reward, that the elephants would not avoid this monoterpene at lower concentrations. From osamine's position on the list and downwards, we start seeing the elephants avoiding the monoterpenes across all concentrations. Osamine is 51% more abundant in avoided plants, and beta-pinene 170%. Similarly, limonene and terpinolene were avoided across all concentrations. Limonene is 200% more abundant in avoided plants, and terpinolene 2,000. However, despite being 320% more abundant in avoided plants, alpha-pinene wasn't avoided across all concentrations. Instead, the elephants avoided it at the lower, but not the highest concentration. A possible explanation for this might be that alpha-pinene, or high levels thereof, is acting as a foraging cue. Wood rats, for example, specifically seek out juniper trees high in alpha-pinene because it's correlated with lower levels of other more toxic compounds. Alternatively, the elephants might be using high levels of alpha-pinene as a foraging cue to decrease search time by, for example, revealing the location of nutritious woody plants. So what are the main takeaways of this study? Well, firstly, the elephants were more likely to avoid the monoterpenes at high concentrations. This can likely be attributed to the increase in post-ingestive costs of toxic compounds like monoterpenes at high concentrations. Secondly, the elephants were more likely to not avoid the monoterpenes that are abundant in their preferred plants at low concentrations. This implies that elephants can indeed tolerate some of these defense compounds at low quantities. And lastly, given that alpha-pinene was not avoided at high concentrations, the possibility exists that elephants might be queuing off of a potentially detrimental compound to facilitate foraging. I would like to thank Adventures with Elephants for allowing us to use the elephants in our experiments, and Recruit and NRF for providing financial support to the study. Thank you.
Thanks very much. We do actually have time for a few questions. Lunch is obviously getting close. Okay, then you can move on and we will carry on with the program. Next up we have Claire Pankey talking about the effect of leaf density on elephant choices between patches.
Okay, so making good foraging decisions is vital, and in order to do this, herbivores likely use a broad range of sensors, including olfaction and vision. So the use of olfactory cues to make foraging decisions has recently received a lot of attention. For example, African savanna elephants make use of olfactory cues to make their foraging decisions between individual plants and patches. However, the extent to which visual cues are used to make the same foraging decisions is less well understood. The few studies that have been conducted have focused on grazers, in particular, the use of sward height as an indicator of food availability, where grazers select the tallest swards. Unlike sward height, tree height does not necessarily relate to food availability. So we suggested that a better measure of food availability for browsers would be leaf density. This is because a greater leaf density will allow browsers to take larger bites and therefore increase their instantaneous intake rate. However, the question still remains, from how far away are browsers able to use visual cues to make their foraging decisions? So to answer this question, we had two main objectives. Firstly, to determine whether elephants are able to distinguish differences in artificial leaf density. And secondly, if they are able to make these determinations, we want to determine over what distances they are able to do so. So with these objectives in mind, we came up with 3 predictions. Firstly, since larger leaf density means more food, we predicted that they will be able to distinguish differences in the leaf densities. Secondly, since visual cues become less reliable with distance, we predicted that the elephant's ability to discriminate differences in leaf density will decrease as distance increases. So that would mean that they are nearsighted. Alternatively, since making long-range foraging decisions would be beneficial, we predicted that the elephants will be able to make these discriminations over a range of distances. So that means that they would have good overall vision. So I know these look like QR codes, but I promise you that they're not. Please do not try and scan them. What these are, they are simulations of bushes. So they are equal in size, but varying in leaf density, including 150 and 25%. So these artificial bushes comprised of black and white patchy circles that were 1 meter in diameter, printed on 1.2 by 1.2 meter canvases. So we used black and white in this case. That's just standard when testing eyesight, and colors only used for color vision, testing color vision. So before the data were collected, the elephants were trained that selecting the larger leaf density would result in a food reward. This consisted of 4 handfuls of game pellets, which is equal to roughly 100 to 125 grams. However, if the elephants selected the smaller leaf density, they would receive nothing. So for the experiment itself, the elephant being tested was instructed to stand with its front feet on a mat, which you can see here. I'm shaking a lot, sorry. And then the elephant being tested was also instructed to point their trunk at this little plate. And this was to ensure that they were facing forward. Then figure B is the experimental design from the elephant's perspective. So there you can see the mat and the plates. Yes, and then so 2 canvases were placed in freestanding metal frames. In this case, it's your 50% and your 25%. And this was to prevent the elephants from potentially keying off any handler standing behind the canvases. Then 2 small orange cones were placed in front of the canvases, 3 meters away from the elephants, and these were used by the elephants to indicate which canvas they wanted, the left cone for the left canvas and the right cone for the right canvas. Then, to give instructions, a handler stood between the cones and the canvases. So once at the starting position, the elephants were instructed to select one of the canvases by touching the cone that corresponded. Once they had made their choice, they were given the respective food reward and asked to move back to the mat. This procedure was carried out over a range of distances, which I will highlight in the next slide. Finally, the distance between the canvases was increased by 0.14 meters for every 1 meter that they were moved away from the elephants. So we tested 4 elephants using all 3 of the possible leaf density combinations. Each elephant was tested 5 times per combination at each of the 6 distances, which included 3, 5, 10, 20, 40, and 80 meters. In addition, the distance of the canvases away from the elephants, the leaf density combination, and the side of the canvases, either on the left or the right, were generalized estimating equations, which takes into account that the data has a binomial distribution as well as repeated measures. The independent variables tested included distance, leaf density combination, and the interaction between these 2, but the interaction was not significant. So with regards to the results, on the y-axis, we have the proportion of the elephant's choices being for the larger leaf density, and on the x-axis, we have the leaf density combinations. The error bars indicate a 95% confidence interval. That dotted line over there, indicates 50% of the choices being for the larger leaf density. Anything above that line indicates preference for the larger leaf density. Below that line indicates preference for the smaller leaf density, and an overlap indicates random selection. So here you can see that the elephants were able to discriminate differences in the leaf densities, and this is in line with one of our predictions. With regards to distance, the elephants selected the larger leaf density at 3 and 5 meters, but they were unable to do this at 10 and 20 meters. This is once again in line with one of our predictions. However, at 40 and 80 meters, things start to get a little wonky. At 40 meters, you can see that they seemingly could select the larger leaf density, but then in contrast, at 80 meters, they preferred the smaller leaf density. So this brings up the question of what could potentially be giving this sort of, unexpected pattern. And with regards to how the mammalian eye works, the results don't really make sense. So most mammals have binocular vision, and this allows them to perceive their environment in 3D, as well as determine the distance of objects. So to determine the distance of objects, the lens of the mammalian eye will change shape. It will thicken to focus on close objects and thin to focus on distant objects. And if this change doesn't occur, this will result in either far or nearsightedness, respectively. So this means that it is impossible for elephants to have good vision when focusing on objects at close and far distances, but poor vision when focusing on objects at intermediate distances. So despite the interaction not being significant, we took a closer look at the results to try and decide why this was happening. So the pattern at 40 meters was seemingly driven by the elephant selecting 100% when compared to 50%, but when choosing between the more obvious comparison of 100% and 25%, they selected randomly. Then at 80 meters, the apparent avoidance of the larger leaf density was seemingly driven by the elephant selecting 25% when compared to 50%, but they once again selected randomly when choosing between the other 2 comparisons of 100 versus 50 and 100 versus 25. So this strange pattern could be a consequence of a small sample size, and increasing the sample size for future studies could provide a clearer pattern. However, despite the odd outcomes at 40 and 80 meters, the overall pattern suggests that elephants have better close distance vision, so that is just beyond what they can reach with their trunks. This ultimately suggests that they use a combination of olfactory and visual cues when making foraging decisions within a patch, but when making foraging decisions between patches, they primarily rely on their olfaction. So in order to gain further insight into the extent to which elephants use visual cues for foraging decisions, we will conduct similar experiments using size and color as visual cues. So for size, we will have a large circle and a small circle of 1 meter and 0.5 meters in diameter, respectively, and that will represent a large and a small tree. And then for color, we will have 3 different colored circles to represent new growth, old growth, and senesced vegetation. Before I answer any questions, we would like to thank Adventures with Elephants for allowing us to conduct our research at their facility. And I'd like to thank you for your attention and your patience. Are there any questions?
Thanks, Claire, for a fascinating talk. We do have a few minutes for questions. So, guys, please. I know lunch is close, but all right, we got some over there. Lawrence and behind you.
Thanks, that's very interesting. Have you considered movement as a way to find food? Not just olfactory. So to explain, so if you move in a certain way through a landscape, you could be more successful in finding food, especially at larger distances. So
yeah, so we sort of like the real world application, I guess, of this research and the olfactory research as well, is to kind of determine exactly how they move through the landscape. So I guess once we know if they, or how they use certain cues, then we can determine this is how they move and that's how they decide what to eat. I don't know if that answers your question.
Fascinating study. Just curious whether you've had the opportunity to do any field testing. So obviously you've now been able to standardize it and objectively. But in watching elephants feed, have you seen any evidence of them using these visual cues?
So the thing is, we tried to remove the olfactory aspect completely because it's so strong and so natural for them to smell something. So it would be difficult to just sort of watch them out, you know, in their natural environment and determine, like, are they using their vision or are they using their olfaction? Because it's very likely that they are using their olfaction in that case. So we tried to remove that aspect.
All right, that's no more questions. Then we can move on. Thank you very much.
Thank you.
And next up and last talk before lunch, we have Bianca Tesling talking about staying on the topics of elephants and how far do African elephants disperse seeds and nutrients.
Hi there, guys. So for those of you who are expecting an item from Beyonce just before lunch, I'm sorry to disappoint Dan and Andrew specifically. But yeah, I'm going to be talking science. So it is kind of generally accepted that elephants can move these super impressive distances in a day. But my study was looking at how far do they actually move and how far do they disperse seeds and nutrients in the process. So I worked with Simon Camille James from the CNRS and Adrian Schrader from UP. So as elephants move across the landscape, they ingest seeds and nutrients. And specifically, the ingestion of seeds and then defecating them away from the source is known as endozoochory. How do seeds and plants benefit from endozoochory? Firstly, we see increased germination success and rates. Firstly, due to being passed through this digestive tract and decreasing the mechanical protection. And then secondly, these seeds are deposited into the world's greatest fertilizer, which is super moist and so full of nutrients. Then, plants being dispersed by these elephants experience faster range shifts, decreased parent-offspring competition from being deposited away from the parent plant, and then lastly, these interactions between metapopulations increase genetic diversity in metapopulations. Africa's savanna elephants can play a specifically large role in endozoochory seed dispersal due to a few key characteristics. Firstly, they have a very diverse diet, ranging upwards from 350 fruit species. I mean, that's quite impressive. Then secondly, their large size allows them to ingest megafaunal fruits, who are specifically dependent on elephants for dispersal. And then their large size gives them a longer metabolic retention time, which could allow them to move farther from the source during their movements through the landscape. And then they have humongous home ranges, which could range up to 2,000 square kilometers. I mean, that's pretty big. And they could then disperse the seeds and nutrients pretty far within these home ranges. So how far they really disperse seeds and nutrients depend on two key factors. The first one is how long they are retained within the digestive tract. And it has been suggested that vegetation is being passed within 21 to 50 hours, while Powell suggested that seed size determines how long it takes for seeds to pass. So small seeds, smaller than 5 millimeters, pass within the first 24 hours, medium-sized seeds within 48 hours, and then large-sized seeds within 72 hours. And then the second factor determining how far seeds and nutrients are dispersed is resource distribution in the landscape, and how this then differs between seasons. So during the wet season, resources are super abundant, and it could allow these elephants to move freely through the landscape and potentially disperse seeds and nutrients very far from the source. Alternatively, these resources that are so abundant could allow them to just move in circles and really not disperse seeds and nutrients that far. However, during the dry season, resources become less abundant. And originally, during the beginning of the dry season, elephants will try to contract their home ranges around water sources, as they are super water-dependent species. So this could result in small net dispersal distances and short trips. However, as resources become less abundant further in the dry season, they are being forced to move further away from their water sources, and this could result in very fast movements between water sources and vegetation, or just between resources, and could result in very far net dispersal distances. Alternatively, they could stay at a source, whether a water source or vegetation source, for longer periods of time, resulting in small net dispersal distances at the source. Sadly, well, luckily for me, research focusing on this is very scarce. So to look at this issue, I used a humongous data set of previously collected GPS data from 33 female elephants from different breeding herds in Wangi, Zimbabwe. During the wet season in Wangi, which is from October until April, water sources and vegetation are abundant throughout the reserve. But during the dry season, which is from May up till September, there's only a few artificial water holes, which forces these elephants to adjust their behavior. So I used generalized linear mixed effect models with my fixed, to determine the total and net distances traveled, with my fixed variables being the season, whether wet or dry and then the metabolic retention time of these seeds would be 24, 48, or 72 hours. And then lastly, I used elephant ID as a random effect variable. So these are my results. Firstly, we are looking at the total number of trips on the y-axis, and then the total distance traveled on the x-axis. So you'll see the solid lines is 24 hours, and then blue is the wet season and orange is the dry season, and then the stippled lines is 48 hours, and the dotted lines is 72 hours. As you can see here, as trip duration increased, the distance of the most common travel distance increased as well, which would suggest that these elephants travel farther during longer periods. We can also see that they are able to travel up to 130 kilometers during these longer periods of time, up to 72 hours, which is quite impressive for an elephant. Then we can look at the same y-axis, the total number of trips, and the net distance traveled. So just a straight line between the starting point and the end point. And here we can see that there isn't really an increase in the most common distance traveled as the duration of the trip increased. Also, here we see that the net distances traveled are a little bit shorter, the maximum that's possible. It's about 70 kilometers. The most interesting part, however, is that the majority of trips, 80% of these trips, were shorter than 10.6 kilometers, although they are able to travel these extreme distances. When comparing between seasons, we saw that on average, elephants disperse seeds and nutrients farther during the wet season compared to the dry season. This difference between 24 hours was significant, with them dispersing seeds and nutrients slightly farther during the dry season. This could be due to a lot of resources around waterholes, so they could stay relatively close to starting point, while the dry season they still have to travel a little bit more to get to their resources because they're less abundant. So within 48 hours, there wasn't a significant difference, and then within 72 hours, due to the abundant resources again, they could move more freely during the wet season, dispersing seeds and nutrients farther, but during the dry season, their movements were slightly constricted. Then, looking within seasons, but between the different trip durations, we saw a significant difference for every single one we compared. These differences, however, isn't much bigger than 1.8 kilometers, and they increase with trip duration, as previously mentioned. So these results are quite interesting. We're looking at the shortest trip first, and we saw that 50% of our trips within 48 hours were shorter than approximately 4 kilometers independent of season. But 1% of our trips within 24 hours was farther than approximately 19 and approximately 21 kilometers. So this is quite impressive. This suggests that small seeded plants from, for example, Grevia species could be dispersed up to these distances. That's quite impressive for a plant. Then, for 48-hour trips, we found that 50% of trips were shorter than approximately 6 kilometers, while 1% of trips was longer than 25 kilometers during the dry season and 31 kilometers during the wet season. These would be large seeded plants, for example, your acacia trees. 50% of trips were shorter than approximately 7 kilometers from the starting point, and 1%, now this is impressive, was farther than 30 kilometers during the dry season and 40 kilometers during the wet season. Now, I don't know about you guys, but I've never seen a tree throw a seed that far. So for them, it's definitely beneficial to be dispersed by these animals. However, it is important to note that small seeds do show low survivability when passed through the digestive tract due to the acidity. In conclusion, it seems that these elephants can disperse seeds and nutrients farther during the wet season compared to the dry season, and that they can disperse large seeds farther than small seeds. So elephants are capable of these extreme distances, up to 125 kilometers, but the majority of these trips result in net dispersal distances up to 10.6 kilometers. Again, from a human's perspective, it's not as impressive as we expected, but from a plant's perspective, this is amazing. So what is next for this study? We want to consider seasonal migrations, when elephants move over the land to reach more beneficial grounds. So a previous study conducted in Wangi by Tipa and his colleagues found that more than 20% of elephants in Wangi have wet season ranges in Botswana, and then they move back to Wangi during the dry season when they're concentrated around a few artificial waterholes. Now, these seasons can differ with up to 260 kilometers, these ranges. So what I want to do in my study now is I want to identify the elephants that do migrate. And I want to see during these migrations, how far do they move within my different trip lengths, and then how far do they disperse seeds and nutrients within these trips. So yeah, thank you so much for your time. Any questions?
Thanks, Bianca, for another great talk. We do have time for some questions. Okay, Sally, you can start.
Thanks, that was really fascinating. I was thinking, I mean, different plants flower and fruit at different times of year. So have you thought about finding out which plants are fruiting during the time when the animals are doing these big migrations? Because those would be the plants that would be most benefited from the dispersal.
Yes, absolutely. We have considered it. I haven't started with that part of it yet, but we did find during the original study that the majority of these plants seed during the core dry season, the beginning of the dry season, but there are some species that seed during the wet season, etc., etc. But yes, these possible far dispersal distances during migrations would only be beneficial to certain plants, but still nutrients are being dispersed during these times.
Okay, Dan.
Thanks, Bianca. Bianca, in terms of, you mentioned the water holes in Wangi, and that was one of the big ecological things there, that they had pumped water holes all year, and you mentioned just a few were operational during your study period. Can you remember, or do you know how many were actually running? Because they seem to all be collected up on the eastern side of the park during your study. How many were active?
So no, I have not considered that yet, but I do know that during the wet season, the majority of the natural and artificial water holes throughout Wangi is working. But during the dry season, like you guys saw in the last slide about what's next, there's this very big concentration of elephants in the northeastern corner of Wangi, and that's where the artificial water holes are concentrated. But it would definitely be interesting to see if one could manipulate where water holes are to be able to move these plant ranges.
Okay, one more, yeah.
Well, during the dry season, it is big enough to keep them alive. But the big thing is Wangi isn't fenced, so they are able to move freely. You wouldn't be able to know if Wangi could facilitate all of these animals except if you fenced them in and then saw what it did to the environment. Does that answer your question?
Okay, I have one final question. I was wondering if you'd considered retention times of seeds in the gut, because that's obviously, it's all very well if an elephant can move over 100 k's in 72 hours, but how long does the seed actually stay inside the elephant?
Well, as Paul suggested, he colored these seeds when they were ingested, and then different feedings had different colors, and then when they came out, he knew, okay, I fed this to the elephant 24 hours ago, and so on. So that's the times that we considered.
Okay, great.
Am I misunderstanding?
No, it's fine.
question?
I'll chat to you at
Yes,
tea or something. Cool. Okay, great. Thank you very much, everyone who gave talks in this session. And just one reminder for everyone who's talking this afternoon, if you can please make sure you get your talks loaded during lunch. And yeah, off to lunch you go, and please be back by 2 sharp.